Hi, everyone. My name is Worskhop, and I currently chair the H3Africa Microbiome Task Force, and as well as the African Microbiome Special Interest Group that we're getting started. Talk more about that a bit later, but just to say we're very excited that you're able to join us for the second African Microbiome Day. This is, like I said, the second iteration of this event. We did have this last year, and it was really widely accepted, and we're hoping that we don't disappoint this year. So we hope that the research we have today gives you a bit of an insight to some of the research going on in Africa. So this is, and just generally in low and middle income countries, just to say that this is not definitely all encompassing. This is for us to talk about some of the research going on. So we hope that this gives a bit of an insight and makes us more curious about the research going on in Africa, and also encourage us to share some of our work. You realize that a lot of these projects are in different stages. So I want to encourage you to next year, hopefully, just share what you have. The idea is to just bring it forward, get some feedback, and hopefully build collaborations. Would like generally knowing fully well that collaborative research is pretty much the way to go nowadays, just to hope that this creates a platform or a bit of a start to some networking possibilities. The format for today pretty much is we're going to have invited speakers, and after the talks, you'll be able to ask questions, 1 or 2 questions. Welcome to type in your questions in the chat box, or at the end, raise up your hands and ask questions if time permits. And if not, if you have burning questions, please go ahead and type them in anyway, and we'll follow up with the different speakers via email communication, and hopefully you get the answers you need. To say welcome again to all our invited speakers and everybody that submitted abstracts. We're very, very grateful and very thankful that you took the time to honor our invitation or took the time to submit abstracts, because without you, this day would not be possible. So just to say a big thank you again. And we're looking forward to an excellent day of research, of networking, of just general communication and interaction about the amazing and the budding microbiome research field on the African continent. We've really tried to bring in speakers from different parts of Africa, but we might not have been very successful, but we are still working on it. So, so hopefully it's a good use of your time the next 4 hours. Just to say as well again that we have a break at some point. And during that break, we're going to have breakout rooms. Should you decide there's somebody you'd like to chat with, you're welcome to invite the person to breakout room just to communicate with the person. And you notice as well that this is not your regular webinar format. We've done this because we would want a chance for everybody to see those that are on the call and get a chance to send messages if necessary. But we hope that your main attention is on the research being presented. So thank you very much again. And we have 3 moderators, including myself for the day. That's 4 in total. And some people are going to be multitasking as both moderators and speakers today. So I'm going to go ahead and get started. I'm not going to introduce her. She can introduce herself. She doesn't need an introduction. For the first session, we have Professor Morenike Folayan. So please go ahead and
thank you, Vogue.
Sure.
And thank you everyone for being here. The first session I'll be moderating has 3 sessions. We have 2 speakers, one at the beginning, one at the end, and sandwiching between our abstract presentations, which you will show as slide. It's indeed my pleasure to welcome the first speaker in the person of Professor Modupe Koka, who will be speaking with us in the next 19 minutes, which I hope would also include 4 minutes of questions. I hope she'll be able to do this in 15 minutes. She'll be talking to us about diseases associated alterations of the oral and gut microbiome in African populations from discovery to treatment. Dupe and our team has been working around microbiome modulations, which we might find very important for therapeutics, including better long-term health outcome, particularly in young children. And she has worked on the microbiome as well as HIV in young people. Definitely a lot of our work is here in Nigeria. And more recently, she has been involved in research related to SARS-CoV-2 epidemiology. It's my pleasure to bring on Dupe, who is also a friend, to spend the next 15 minutes speaking and 4 minutes to take questions. Thank you. Over to you, Dupe.
Wow, that's a wonderful. Thank you so much, Prof, for that. Such a pleasure to be here. I'm going to try and share my slides as I introduce myself again. So just to remind you, I am a dentist by training, so many of the things that you're going to be seeing or hearing about really are in relation to, can you see my slides? Or should I
Yes,
stop?
please. It's very clear. Very clear.
Is it a presenter mode or is it in a
Presenter mode.
okay? I must see
We're
what
at the
I'm
very beginning now. Yeah.
okay, good. All right, perfect. So I'm a dentist by training, as Prof has introduced me. I am also an epidemiologist. So you'll be getting a feel of this presentation from a very holistic. I'm not going to be zoning on my own work, although I'll mention some of my work, but I really wanted to ginger everybody. This is my goal, ginger everybody to think about not just the gut, which I know most people on this call are going to be studying, maybe a few of others in other niches, but also look at the oral microbiome. My next assignment is really to ginger you to really, we have a clarion call to work with African populations to try and study our conditions, things that affect us primarily, and then kind of see the trajectory from discovery, prediction, and then treatment. These are my contact details should you want to reach out to me. I'm very open to collaboration. I have no financial interest to declare, so as I go through some of these work, there's nothing that I benefit from this sharing some of the things that I'm doing or things that I find. This is the outline that I will try to work with. Again, 15 minutes is short, but I would introduce, which I don't need to, introduce to the impact of microbiomes in health and disease, share some knowledge gaps in African populations, and then kind of give you some research examples or applications from discovery, prediction to treatment, and then share my thoughts for future directions. All right, so we know, and this is not news to anybody here, that the microbiome is very important. It's typically called the super organ, right? Where you see, we know that we have 10 times more cells, microbial cells, than human cells. So they're over and around us, right? They're encapsulating everything that we do. But just keep in mind that we're not talking about just the microbes, but also the genome that encode them, and not just what the microbes are, but what they're doing and what they're producing and how they're making things function and how they contribute to health and disease. So I want to just keep that in your mind as we go through. And there are many ways we can study the microbiome. I'm not going to go into detail, but sometimes you will hear through the course of this seminar, this workshop, going through some ways people talk about metagenomics, metatranscriptomics, metabolome, but you can look at different things. What are there? What microbes are there? What are they trying to do? What is making it happen? And what are the end products of these activities? As you must have heard from Prof's introduction, I work primarily with the children, but I'm not limited to working with adults. Or in fact, I see a very similar trajectory when children are developing microbiome and then aging microbiome, almost like the two ends of the spectrum, right? So you see over time, a child is born and they're inundated by several exposures, and then they have increasing abundance and diversity. And then we know things that change them. We have age as a very strong factor. We have ethnicity, diet, dentition, as I'll talk to you a little bit about, and medication, smoking, disease factors, as well as genetics. We understand that, especially for the gut microbiome, there are very strong factors that drive it. And I've been privileged to be part of a US-based cohort, which has kind of helped me to build on an African cohort, which is very exciting. But to think about it, that we know that delivery mode, the kind of delivery that happens for the child matters. The dentition, the diet, then the dentition, breastfeeding or not breastfeeding. We'll talk about that. Some of you are going to be discussing breast milk microbiome. And we know that over time, when the child becomes like 2 to 3 years of age, there's some plateauing. The microbiome begins to look like an adult. Well, again, I'll share with you my hypothesis about that. But as you see from the right slide, the diversity increases with age, but the inter-individual variability also reduces, depending on who you're comparing, right? If you're looking at the population, then there's less inter-individual variability with age. But if you're comparing that population to another population, of course, there's a huge range of microbes that you will see. We all know this sentinel study. I don't want to belabor you because I don't have a lot of time, but just to kind of drive home the fact that the gut microbiome has been implicated in supporting or contributing to obesity. And these are sentinel papers that were published looking at fecal microbiome that was transplanted into recipient mice. So these microbiomes can actually infer or induce similar phenotypes into the mice. And what is interesting also is that even those who are receiving the lean recipient mice can also transfer to the obese recipient mice if they're receiving the same, if they're receiving low-fat, high-fiber diet. So these are kind of very interesting findings that have made us kind of interested in, but I want to ask you what you know about the developing oral microbiome. The oral cavity is the beginning of the gut, and being a dentist, it's something that I know that before, but it's not something that I've seen being studied extensively, especially in this field of microbiome. What I've found interesting is that the same things that perturb or that drive development of the oral gut microbiome also drive development of the oral microbiome. The same things I mentioned to you before. And also to kind of add to that, some of my work has actually seen that with age, the microbiome increases, diversity increases, but also plateaus, the same way it plateaus with the gut, also plateaus with the oral microbiome. So this is a salivary microbiome using 16S RNA sequencing. And you can see here that there's once it's about 3 to 36 months of age to like 72 months of age, there's a plateauing of the microbiome. And my hypothesis is that there's dentition. Dentition is complete. The diet, of course, changes. The way they masticate food changes, and it also makes it easier for it to also change the gut microbiome. So there's a lot of relationship with the gut and the oral cavity. I also want to drive home the point, you may already know about the gut and how it's important in driving immune factors and supporting health, but do you know about the oral microbiome and how there's a lot of emergent evidence connecting the commensal microbiome or the opportunistic microbes that are in the oral cavity, how they also infer or support or drive disease systemically. And we have so many examples in this slide, but I wanted to highlight some of them that maybe have been really important in the past decades. For example, the cardiovascular disease, as well as the colorectal cancer. So specifically, FNUC has been seen to be implicated, and I'll share some slide with you, implicated in colorectal cancer. So it's a typical pathogenic bacteria in the mouth, but when it's seen in the colorectal tissue, it facilitates disease. So with that, this is what I was trying to say. So this is what I wanted to kind of introduce to you, that really the local piece is there. Also, there's a systemic piece in terms of the impact of the oral microbiome. And I want to just walk you through this beautiful work that was done by a team and published in Science Journal, and really looking at the fact that the oral microbiome is complex, right? You have different niches in the mouth. There's so many sites beyond just the saliva. So I'm looking here at the supragingival biofilm, and you can see how the architecture is very different from the tongue. Specifically, you can see that the Corynebacterium is like a filamentous filament and is covered by strep. While in the tongue here, you have typically what they call nitrate-reducing bacteria, Rothia and Neisseria kind of intermingled here, and is very important for helping support cardiovascular health. When you look at systemic disease, or in terms of non-oral sites, you see another similar, or another important factor. Two things have come out, some of the work that people have done, especially in the Western world, looking at the impact of Fusobacterium nucleatum and how it activates signaling pathways that allows for the proliferation of colorectal tumor cells. Similarly, P. gingivalis has been important in targeting proteins that cause chronic inflammation in rheumatoid arthritis. So again, these are all studies that have been done in the Western world, but not quite. The question is, what do we see in the African populations? That kind of takes me to the next question. Do you know that most of the studies that we have right now in Africa are typically, well, you have a lot of brain, urogenital, and then you have very few in the gut and the oral. Gut is still predominantly more, but the oral is less. So you can look for the blues. You can actually scan through and looking for the blues in the mouth, or even the nasopharynx, right? But even this, look at the sizes. These are greater than 10. I show you another slide here of just another beautiful review by a group looking at different studies conducted with the gut microbiomes. But I didn't find anything with the oral, so maybe that's something somebody here or will work on. But when you look at the work that we have been doing for example, in sub-Saharan Africa, even they have different groups and diseases, right? We have the classical dental caries, some of them have perio, some of them have diabetes and cancer, breast cancer, but it's nothing close to the gut microbiome. But why you can ask, why are we particular about studying the African microbiome? We've learned a lot with the Western world. Why do we need to study African microbiomes? So look at this slide. With reducing national income, the kind of microbiome is very different. In fact, the problems that we're seeing, while there's a transition from infectious diseases to non-communicable diseases, we see clearly that the microbiome is not the same. There's a different diversity, especially with different ethnicities, diet, industrialization, genotypes. They derive some of these findings, and some of the findings that we're seeing in the Western world may not apply in the African populations. Also, when you look at the UN 17 Sustainable Development Goals, if you look at each of these 17 ones and you transpose them and think about how you can interrupt or modify or change the trajectory, you see a very nice overlay with microbiome being critical to meeting each of them. And I won't bore you with going through all of them, but there's a lot of work that we can do to kind of keep those sustainable goals in our minds, and using microbiome as a tool to support meeting these goals. I wanted to bring an example that was very interesting in my mind, looking at what I was trying to explain about the African population. While we know that there's a wide diversity, even within the regions, there's so much variability. We have those who are hunter-gatherers, agriculturists, and those who are industrialized. So we need to start thinking about how we keep in mind that while we are studying these populations, they are inherently different. And I share with you some of the work people have done with hunter-gatherers, and some people here that are actually on this call have really done a lot of work with urban and rural differences with the gut microbiome, and just kind of highlighting the big problem that we have coming up, which is antibiotic resistance. So with this, I want to now kind of dive into some examples. So when you think about moving from discovery to treatment, the big things are the trajectory. We discover to understand, natural history, what's happening, what ways we can use to monitor our populations. We can also try and get biomarkers that could be predictive and help us to target those who need interventions. And so therapeutics is the next. So you have this nice flow from discovery, prediction, and then therapeutics. And I share with you some of these ideas, but by no means exhaustive, and I'll go through them a little bit later. But just to keep in mind that you cannot work with therapeutics until you've really had a handle on discovery, and discovery within our population, and within the things that really are prevalent in our region. So I want to share some of my work. I'm a dentist, like I said. I'm privileged to be working in Nigeria, where I come from. Although I'm actually trained as a dentist in Nigeria, but also trained in the US as an epidemiologist. And working with the microbiome has really been an eye-opener for me. It's like a tool. So think of it like if you're looking at a problem, in my case, HIV and dental caries, the tool here was the microbiome. And how did this come about, you may ask? I, being a dentist and also working with public health programs in Nigeria, I was privileged to work within the University of Benin Teaching Hospital in Nigeria, within a CDC-sponsored project where they were already receiving therapy. Most people were receiving therapy. And one of the key things we found, and interestingly, was that most of these individuals were already on HAART, and they were still getting dental caries. These are kids who are born to HIV-infected mothers. So you have these three groups of children: those who are HIV-infected. We have another group that are HIV exposed but uninfected, meaning that their moms were HIV infected, but they didn't acquire the virus. The other group is neither exposed or neither infected. So we had that opportunity to observe them, and we saw that consistently they had a higher prevalence of caries. So the question was why? And we were privileged to look into the salivary microbiome to kind of pass out these three groups. And we found that HIV-infected group was similar to HIV-exposed but uninfected group. But the microbiome for these two, HIV-infected and those who are neither exposed nor infected, were different. And then the HIV-exposed group was also different from the HIV-unexposed group. And we saw very important, sentinel papers that also found things that we found, like example, like trachea, seem to be very protective of the microbiome, trachea as well as Rothia. These are commensal bacteria, but if you have less of that, then you are less likely to have caries. So one of the things that we did was that, we found that was great, but does that help us to know what to do with these kiddos? So we started looking at them at an age where they have mixed dentition, right? A larger population, 4 to 11 years of age, about 568 in this population. We looked at them over time from recruitment to 12 months later. So we have recruitment at baseline, 6 months, and then 12 months later. So we're collecting samples from them, supragingival plaque, as well as other samples, but the samples I'm going to share with you today are really supragingival plaque. This is data that also comes from the same cohort, highlighting the differences in dental caries with respect to the 3 groups, and also differences in growth. Again, these HIV-infected individuals or HIV-exposed children, they seem to have a lower ponderal growth, and they seem to have higher risk of caries. So I work with an amazing team collaborator, Dr. Richards, who also works with me. So instead of looking at the 16S RNA sequencing like we did last time, we focused on RPROC, which is better with respect, in our experience, respect to resolution at the species level. And so we were able to analyze these plaque samples from these 3 groups of children, and I want to walk you through some of the results. So as you can see here, on the left, there's left panel, middle panel, and the right panel. I want to explain to you what this is. This is when a child does not have caries, and the plaque was collected from a caries-free tooth. When they do have disease in animal, and when they do have disease in dentine. And walking you through this, you can see clearly that the separation is stronger when they have disease. So HIV, yes, is a driver, but only a driver when there's dental caries, when there's disease. Furthermore, we went on, so Dr. Mann is really the postdoc that drove this research. I just want to acknowledge her. It's her work that I'm presenting today. Also, to kind of highlight the fact that we're expecting, you may know that there's a bacteria called S. mutans, which is well known to drive dental caries. And we're expecting to see it abundantly in HIV-infected individuals, those children who were born and acquired the virus from their mothers. And surprisingly, we didn't see it. We saw a lower abundance of this carcinogenic bacteria in HIV-infected children compared to those who were either unexposed or uninfected. And so one of our hypotheses, if you look at this closely, you look at the people that don't have S mutants, they look very much like healthy teeth or healthy profiles from healthy teeth. And our hypothesis was really like maybe there's a natural history, there's an upsurge and there's a natural decline, as you can see here. We lose out resources, the data, the bacteria has done their work, and then there's a decline in abundance. So really kind of takes us back to why we should not just be looking at taxonomy, but we should be looking at functional abundance, as well as the metabolites or the byproducts of activities in the oral cavity or even the gut.
We'll have just a minute to go. I hope that's okay.
Yes.
Thank you.
So I just want to quickly walk you through prediction. So another piece of prediction is also looking at how we can predict those who acquire the virus. And I have a project here. You can ask me about this later, looking at whether or not HIV individuals have a higher risk of acquiring oral HPV infection using machine learning. So with respect to therapeutics, and I'm sure we're going to hear from Dr. Gordon, amazing about this, looking at several ways of modulating the gut microbiome or even the oral microbiome. But little has been done with respect to the oral microbiome because of the fact that we don't have enough discovery, enough prediction, really. We know stuff like Lactobacillus reuteri has been implicated as a good probiotic for caries, but very few studies in Africa. These are all Western countries. All right, so I just want to end by saying that the oral and gut microbiome, there's a relationship between these two. We should be studying them together. And there's a crosstalk.
Let me just stand on when I start.
And then there's a discovery that has to happen before prediction and therapeutics. I also want to highlight the need to move from correlation to causation. As you may have seen for many of these studies, they were just looking at associations. And when we focus on our own population, we should begin to think about using cultures, using animal models to validate what we're seeing in these human studies. I also want us to refocus, and we're going to talk about this, I'm sure, with Dr. Gordon, but really refocus our mind on our own needs in African population. There's malaria, there's lower respiratory tract infections. There even TB is still a problem. HIV, yes, has been for a while, but I think we should begin to shift our minds, develop a biorepository, think about collaborative networks, invest in bioinformatics and data science approaches and think about a holistic and integrated multidisciplinary plan as we try and address some of these research questions. And think about personalized medicine as an adjunct to public health. We cannot begin to think about only microbiome as a tool without thinking about how we can use it as an adjunct to public health. So with that, I just want to say thank you for listening to me. I have a lot of people that work with me. These are people that I worked in Nigeria with, as well as those in the US. And it takes a team to get the work that I do. Thank you for listening.
Thank you very much, Jupe, for that brilliant piece. And yes, acknowledging that it takes a town in Africa to get anything done. I also love the way we ended around the issue of personalized medicine, because that would definitely take us very well into Professor Kundi's presentation, where she does a lot of studies around personalized medicine. Please, if you do have questions, send them into the chat box. We're going to also have another session where we're going to take one-on-one responses. But in the interim, we'll have to move on to the next sessions where we'll be looking at the slides. How do we proceed on this work? I think we'll have the slides presented all together, the 5 slides presented all together, and then at the end, we'll also take questions. Please do not leave Professor Coker. You would definitely have questions to respond to. Thank you very much. All right, please let's have the presentations. We do have 5 slides, abstracts as flash presentations. We'll be taking first Samantha Snell's presentation on exploring the interaction of host genetics and gut microbiome in obesity in an African population, and we'll go down the slides that way. Over to the organizers.
Hi everyone, my name is Sam, and today I'll be sharing my master's research, which was titled Exploring the Interaction of Host Genetics and the Gut Microbiome in Obesity in an African Population. Obesity is a highly prevalent health concern that is on the rise in sub-Saharan Africa. Regarding the etiology of obesity, human genetic variation and gut microbiota have been implicated. Due to the polygenic nature of obesity, genetic etiologies are best assessed with polygenic risk scores using GWAS summary statistics for obesity-related traits such as body mass index. Even though these 2 factors have been implicated in obesity, it has been independently, and little is known about how they interact. This has been previously explored, but not extensively, and not in the case of South Africa. Therefore, the aim of the study is to identify possible associations between host genetics using polygenic risk scores as a proxy, BMI as a measure of obesity, and gut microbiota composition in a female African population. The study cohort comprised 169 female Africans from Agincourt and Soweto who were enrolled in AWIGEN Phase 1 and microbiome pilot studies. For the purpose of the study, they were classified based on their BMI. Using SNP genotype data from AWIGEN 1, polygenic risk scores were calculated using summary statistics from 3 different studies with 3 different ancestries. The most predictive polygenic risk score was then used for downstream analysis. Participants were ranked into deciles. The first 3 deciles were those of low polygenic risk, who were controls, while the last 3 were those of high polygenic risk that were cases. The intermediate deciles were excluded from further analysis. Microbiome analysis was then performed using corresponding 16S rRNA gene sequencing data. A range of microbiome analysis was performed, including data processing, quality control, taxonomic assignment, rarefaction analysis and richness estimates, alpha and beta diversity measures, and differential analysis of taxonomic abundance. Through this, taxa of interest were identified. Finally, possible interactions were then assessed using linear and multiple linear regression models. The multi-ancestry meta-analysis, or MAMA-PRS, was the most predictive in the study cohort. It produced the highest PRS R-squared, meaning that approximately 6.65% of the variability in the BMI phenotype can be explained by host genetics using PRS as a proxy. Additionally, MAMA produced results that were of high statistical significance. Therefore, it was selected for decile separation and for further downstream analysis. Moving on to the microbiome analysis, comparing those of high and low genetic susceptibility to developing obesity. There was a lack of association identified in the alpha diversity measures and polygenic risk. The low polygenic risk group had greater alpha diversity, with the greatest difference being the Shannon Diversity Index. This suggested that there is greater species diversity within the low polygenic risk group. Regarding beta diversity measures, there were community differences that were observed between groups, but definitive conclusions could not be made. When comparing taxa at a genus level, the polygenic risk groups were further divided into high and low BMI subgroups. Participants with high risk and high BMI had the greatest abundance of Prevotella 9 compared to their low risk, low BMI counterparts. The predominant genus in the low risk, low BMI subgroup was Bacteroides, and it was found at highest abundance in all the subgroups, and the high risk, high BMI subgroup had the lowest abundance of the same taxa. These abundances suggest possible associations, therefore making Prevotella 9 and Bacteroides taxa of interest. By identifying taxa of interest that discriminated between lean and obese or overweight groups, as well as high and low polygenic risk groups, overlapping taxa could be identified. It was found that Vadin BE97 was overrepresented in both the overweight and obese groups, as well as the high-risk group, making it the third taxa of interest in the study cohort. Therefore, there were 3 taxa of interest identified. However, when regression models were run to assess possible associations, only one was of statistical significance, and this was with Prevotella 9. This was suggestive that there was an interaction between BMI, polygenic risk, and Prevotella 9, as well as suggesting that there were other factors influencing BMI in the study cohort, other than host genetics. Previously, abundances of Prevotella 9 have been associated with obesity, and there have been reports that host genetics influences abundances of Prevotella 9 as well. It is important to note that due to the small sample size and subsequent lack of associations that could have been identified, this study should serve as a model for future research exploring relationships between host genetics and gut microbiota. Thank you so much for your time.
Thank you, Samantha, for the illuminating lecture, and we'll look forward to questions at the end of the session. We'll now have the next presenter, Lara Dugas. Let's have the slide, please. So Lara is presenting on gut microbiota and fecal short-chain fatty acid differs with adiposity and country of origin, the METS microbiome study. Thank you. All right.
Dr.
Oh, sorry.
Lara.
We're skipping Lara.
No,
Okay.
it's Matsepo and then Lara.
Okay, sorry, I have a different order. Please go ahead.
Changes in the gut microbiome environment link urbanization of South Africans to increase in risk of non-communicable diseases. Non-communicable diseases, NCDs, present a primary health threat in high-income countries, but are now increasing in developing countries. There is a widely supported hypothesis that changes in diet and lifestyle associated with urbanization as part of the economic development in sub-Saharan Africa are the main drivers of NCD risk. However, the underlying biological mechanisms are not fully understood. When we eat food, it goes through digestive tract, where it is digested by enzymes and soluble nutrients are absorbed. Any remaining undigested food material ends up in the large intestine, where it is metabolized by resident gut microbiota. What is intriguing about the gut microbiota is that it has large amount of genetic material and has high metabolic capacity. As a result, remains of various dietary compounds are metabolized into diverse metabolites. Blue arrow is pointing at short-chain fatty acids produced from fermentation of fiber. Short-chain fatty acids such as butyrate has been found to be protective against the development of colon cancer. Red arrows are pointing at various nitrogenous compounds and secondary bile acids that have been associated with dysbiosis, inflammation, and development of many diseases. We hypothesize that the transition to high-fat diets promotes dysbiotic traits of the microbiota reflected by the suppression of butyrate-producing bacteria and increased levels of bacteria that produce tumor-promoting secondary bile acids, as well as the distinct patterns of bacteriophages. We aimed at investigating how the ongoing transition from traditional to urban lifestyle affects the gut microbiota and its metabolome, by comparing between the fecal bacteriome and virome of the rural and urban Xhosa people. Our study is a cross-sectional observational study based in Cape Town, a city that for many years has been receiving migrant populations from outside the country and within the country. Majority of those from within South Africa come from the Eastern Cape province, hence we set up a rural cohort in the Eastern Cape and urban cohort in Khayelitsha, and we recruited a total of about 45 healthy middle-aged men and women. Dietary assessment was done. Various clinical samples were collected and analyzed. Of these, the fecal samples were analyzed for bacterium and virome, and the resultant sequencing data was characterized for composition, diversity, and abundance. Bacterial alpha diversity was higher in rural than urban cohort. The two clusters of samples indicate that both cohorts have distinct bacterial composition. Similarly, the two cohorts have distinct differentially abundant genera. The red arrows are pointing to Fusobacterium and Bilophila, which in many studies have been associated with colon cancer and bowel inflammation. The blue arrows are pointing to Treponema, Faecalibacterium and Prevotella, which in many studies have been associated with healthy rural lifestyle. Faecalibacterium and Prevotella are known short-chain fatty acid producers. Of the 900 viral particles, less than 200 were shared between rural and urban cohorts. There was no viral diversity between rural and urban cohorts. The samples tend to cluster by their origin, indicating that the rural and urban might have distinct viral composition. Several differentially abundant viruses infecting specific bacterial tags were detected. The genus Bacteroides seems to be the most susceptible to viral infection in both cohorts. Faecalibacterium is targeted by viruses in the urban cohort. This could be a way of controlling its levels, as it was not abundant in the urban cohort. Overall, the role of viruses and their interactions with bacteria and human cohorts requires further investigation. We also note that the differential abundance of tumor-promoting bacteria and the diminishing of short-chain fatty acid-producing bacteria in the urban cohort is very concerning. In conclusion, taken together, these results and those presented elsewhere provide evidence that changes in the dietary pattern, the gut microbiota and virome with urbanization may be predisposing to the development of colon cancer and other NCDs. Further studies are required to determine whether the restoration of specific components of the traditional diet will stop the accelerating rise in NCDs in South Africa. Thank you.
Thank you so much, Matsipo. Very beautiful piece, and quite a lot of questions. Samantha, please note you have questions in the chat box. Please check. And you do have a new question also, Matsipo, in the chat box. If you could answer those questions, we'll be able to catch up on time. I would like to now ask for Lara Dugas' presentation. Please go ahead. Thank you.
Good day. My name is Professor Lara Dugas, and I am the AXA Research Chair for Non-Communicable Diseases in the School of Public Health at the University of Cape Town and an Associate Professor of Epidemiology at Loyola University Chicago. I'm going to be presenting my research titled Gut Microbiota and Fecal Short-Chain Fatty Acids Differ with Adiposity in Country of Origin, the METS Microbiome Study. So the METS study is actually the Modeling the Epidemiologic Transition Study. The study was started in 2009 by Dr. Amy Luke. Loyola University is the coordinating center. The initial aim was to understand the factors which contributed to cardiometabolic risk and diabetes in 5 African origin populations at different stages of economic growth. The 5 sites are situated in Ghana, in Nkwantakesi, which is a rural village about 25 kilometers outside of Kumasi, in Khayelitsha, which is a peri-urban informal township as part of the greater Cape Town metropolis, in Kingston, Jamaica, in Victoria, Mahe, based out of the Ministry of Health, and then in Maywood, Illinois, which is a suburb on the western border of Chicago. What we found in the original MET study is that obesity drives disease. And so we can see that obesity is lowest at 10% in Ghana, and in 2009 was highest among the US participants. Diabetes follows obesity, and you can see that the risk for type 2 diabetes, or the prevalence, was lowest in Ghana and highest in the US. Hypertension was always lowest in Ghana and highest in the US. We were able to understand what people were eating through 2 24-hour recalls. And from that, we learned that the Ghanaians consume a very high amount of fiber at almost 25 grams per day. And that was almost twice as much as the participants in the US who were consuming 14 grams per day. I was very interested in this because we knew that the fiber is digested by the gut microbiota to produce short-chain fatty acids. So my colleagues and collaborators, Dr. Brian Layden from the University of Illinois in Chicago and Dr. Jack Gilbert from the University of California in San Diego, first set out to review the literature to see what international studies have been done looking at an obese microbiome across international populations. And we found back in 2016 that there were only 5 studies that had leveraged international cohorts. And so we got funded to leverage the METS cohort to specifically focus on gut microbiota, adiposity, and type 2 diabetes risk. And what we discovered is that country of origin predicts the diversity of the gut microbiota. On the left, you can see here, and the countries are ranked by the Human Development Index, with Ghana having the greatest diversity in the microbiota, and the US participants and Seychelles participants having the lowest diversity in the gut microbiota. More so, though, we found that whether you were obese or not obese predicted the diversity of your gut microbiota, with those who were not obese having a greater diversity in their gut microbiota. Overall, there were 23 common bacterial strains that were associated with obesity across our 5 sites. The gut microbiota are also able to predict obesity and type 2 diabetes. They can predict obesity with about 65% and type 2 diabetes with about 63%. And this is when we look across all the sites. Country and fiber intake obviously predicts the levels of short-chain fatty acids, and it wasn't surprising because the Ghanaians eat so much fiber that they had the highest amount of fecal short-chain fatty acids, almost twice that of participants living in the US. Your fiber intake corresponds with your fecal short-chain fatty acid, which are actually correlated then with the diversity of the gut microbiota. And when we look at all the 4 short-chain fatty acids and we pool them, we see that Shannon diversity is significantly associated with the composition or the diversity of the gut microbiota. I'd like to thank my funders. This work is funded through the National Institutes of Health as well as the AXA Research Fund, and I'm further funded by the Wellcome Trust.
Thank you so much, Lara. That was really beautiful. We heard from Lara about country differences affecting the microbiome, and we had heard earlier from Atipo about even within countries, you do have diversity between urban and rural areas. Both of them are linking it all to diet. I mean, this is really enlightening. Thank you so much. We'll call on the next speaker, Natasha Shane, who will be talking about, all right, I think I have a very different order. Please go ahead with Dr. Buzi, who will be talking about composition of salivary microbiome in coronal patients from North Africa. Over to you.
Hello everyone, my name is Philippe Bouzid. I am a postdoctoral researcher at the Laboratory Efficiency of the Center of H3Africa in Tunisia. Today, I am really pleased to present our research project entitled The Composition of the Salivary Microbiome in Coronary Patients from North Africa. In our lab, we are interested in the human oral microbiota, which are both the second most abundant and complex microbiota after the gut microbiota. Interestingly, the oral microbiota is stable but remains susceptible to alteration due to infections or other stresses leading to oral microbiome dysbiosis, and this imbalance can lead to systemic inflammation affecting the whole body. Based on previous studies, oral dysbiosis was associated with an increased risk for cardiovascular diseases through systemic inflammation triggering cardiovascular events. In this context, our projects aim to compare the oral microbiota composition between Tunisian patients with coronary artery disease and control subjects to identify a potential pathogenic signature associated with CAD. So the global strategy of our projects required the recruitment of coronary patients and control subjects with the same inclusion criteria, the collection of red saliva and DNA extraction, followed by V3V416S RNA metagenomic profiling of salivary microbiomes on MySeq Illumina platform, and finally a deep bioinformatic and biostatistic analysis. In the first step, we determined the composition of the oral microbiota. At the phenome level, the oral microbiota of patients and controls was dominated by 6 bacterial phyla, and this observation was in accordance with the current knowledge of the oral composition as reported by the oral microbiome database. In our study, we noticed that the oral microbial communities in patients and controls are represented by similar global core oral microbiome. Interestingly, using variable selection by elastic regression model, the results identified a single Echinella as the most influential for CAD status, with a higher abundance in CAD patients, particularly at early stage compared to the control group. From the literature, we knew that the genus Echinella is a periodontal pathogen implicated in infected endocarditis and was detected in atherosclerotic plaques. Also, a pro-inflammatory effects of Echinella was reported. Then, this periodontal pathogen could be a major contributor enhancing the atherosclerotic process by inducing endothelial inflammation. On the other side, apart from bacteria, we unexpectedly detected the presence of the alkaline genus Methanobrevibacter in 3 samples at very low relative abundance. We then performed a deep characterization of methanogens in Tunisian adult saliva using a polyphasic molecular approach for accurate identification and quantification. We identified a high prevalence of methanogens of 81%. We noticed that quantitative real-time PCR was the most efficient methodology, while targeted metagenomic approach was the least sensitive method to explore the archaeal community in the oral microbiota. So through our studies, we characterized the oral microbiota in disease and how the individuals recorded FESTA in North Africa. Interestingly, we detected a different abundance distribution of the genus Echinella in all samples of coronary patients compared to controls, and our findings support the impacts of periopathogens of the oral microbiome that may translocate from the oral cavity to another body site, where they may contribute to coronary artery disease through an inflammatory process. We also reported a high prevalence of methanogens in cellular samples from North African population, and we also noticed that universal approaches using metagenomic or standard 16S RNA sequencing still overlook the human archaeome and partially fail to picture the diversity of archaeal signatures. For more details, I invite you to read our recent article published in Bioscience Reports. At the end, I would like to thank the organizers for selecting my project for our presentation. I also thank the efficiency team who contributed to this project, and surely you for your attention. Thank you.
Thank you very much. Thank you very much. Thanks for contributing to this wonderful discussion. Once again, I want to encourage the past presenters, please check the chat box. And for those questions you can continue to answer, please do. Now we'll call on Zili Maso, who will be presenting the slide titled Discussing Identification of Distinct Cardiotonic Steroid Pulses. Please go ahead with the presentation. Thank you.
Hello, my name is Jeffrey Candy. I'm from the Department of Surgery at the University of the Witwatersrand. I'm presenting work of 2 MSc students, Zili Maso and Hanif Muller, who contributed equally to this work. Ms. Maso undertook the initial HPLC MS development work at the University of Pretoria under the guidance of Professor Duncan Cromarty. Ms. Muller undertook the rat work and blood pressure measurements at Wits University, and the bile acid and cardiotonic steroid measurements with the assistance of Shimatsu, South Africa at the University of Johannesburg. Cardiotonic steroids are modified at position 17 with either a 5-membered or a 6-membered lactone ring, which are found in cardenolides and bufadienolides respectively. Cardiotonic steroids are being used to treat heart failures and arrhythmias. They've been identified in urine and blood and are produced in the adrenals and pituitary glands by bile acids. Cardiotonic steroids bind to the alpha subunit of the sodium-potassium ATPase pump to affect the activity of this ubiquitous transporter. 3 sodiums are pumped out in exchange for 2 potassiums coming in to create an electrochemical gradient which facilitates various cellular and biochemical processes. Different cardiotonic steroids are associated with the different manifestations of cardiovascular disease and are now being increasingly investigated as anti-cancer and antiviral treatment. Although urine has been described as the major route of excretion of cardiotonic steroids, bile has also been identified as a major route of excretion. And therefore it's surprising that cardiotonic steroid profiles have not been determined in fecal material. So we use various rat models, spontaneously hypertensive rats and their ancestral Worskhop coyote rats, measuring tail cuff blood pressure measurements. The rats were transferred to metabolic cages overnight to collect fecal material, which was freeze-dried, extracted, cleaned up, and subjected to HPLC mass spectrometry to identify them. And we also got preliminary data from humans. This slide shows that a whole profile of cardiotonic steroids may be extracted from human fecal material, and this is dependent on the solvent that was used in the extraction process. Each color here represents a different rat strain or different intervention, from control rats through to exercised rats and rats on different diets, and in particular the salt-sensitive rats on a salt diet. So again, in the rat models, we see profiles of different cardiotonic steroids. Finally, cardiotonic steroids were measured relative to total bile acids determined in the WKY rats shown here in pink, and in the rats with high blood pressure, the spontaneously hypertensive rats. And you see a very, very different profile of bufalin concentrations and digitoxin concentrations between these two rats. In conclusion, this is the first report of cardiotonic steroid profiles in fecal matter. The analysis of cardiotonic steroids is problematic from the extraction, the multiple compounds which are identified with identical molecular weight, and the lack of standards. Profiles of cardiotonic steroids in fecal material varied with diet, treatment, and the rat strain. Cardiotonic steroids were identified, bufalin in spontaneously hypertensive rats, and very differently, digoxin from WKY rats. Oben was not identified in any sample, and this is in contrast to data that's been produced previously. And these data suggest that the gut microbiome may be an important variable in the formation of the variety of indigenous cardiotonic steroids produced, as well as in the transformation and metabolism of such cardiotonic steroids. Thank you very much. Questions?
Yeah, I got distracted looking at the excellent conversation going on in the chat box. Thanks, please keep this going. Our next presenter will be Natasha Kishin, who will be presenting as stated in the slides. Let me not waste your time. Please go ahead. Thank you.
Hi, I'm Natasha, and today I'll be discussing some of my PhD work, which investigated the gut microbiome in relation to fetal alcohol spectrum disorders. Fetal alcohol spectrum disorders is an overarching term describing 4 diagnoses along a spectrum of increasing severity consequential to prenatal alcohol exposure. Despite the fact that prenatal alcohol exposure is one of the most preventable causes of birth defects, developmental disorders, and intellectual disability, South Africa has the highest recorded prevalence of FASD worldwide. The Western Cape has the highest documented prevalence of FASD, with approximately 16 to 31% of children affected, compared to the global prevalence of less than 1%. FASD therefore constitutes a serious health concern and a significant social and economic burden. Not every child with prenatal alcohol exposure goes on to develop FASD. In fact, FASD pathophysiology remains unclear despite the identification of both genetic and epigenetic influences. Researchers have also failed to yield susceptibility estimates or identify reliable FASD biomarkers. Although many avenues have been explored, researchers have overlooked a wealth of genetic information that may modulate FASD development, namely the gut microbiome. Neurodevelopment is suggested to be modulated by the gut microbiome via the microbiome gut-brain axis. Fetal bacterial colonization plays a significant role in neurodevelopment. Therefore, abnormal gut colonization during the first weeks of life can alter crucial functions of gut microbiota and can increase susceptibility to disease. Furthermore, neurocognitive development in the early postnatal environment is highly dependent on microbial composition and the corresponding microbial metabolic outputs. Therefore, alcohol-induced microbial alterations may increase the risk of FASD development in infants. We therefore aim to investigate longitudinal changes in infant gut microbiome composition in order to identify a stable gut microbial signature associated with FASD development. Pregnant women were recruited from antenatal clinics in Robertson and Wellington, and stool samples were collected from their infants at 6 weeks and 9 months of age. Active case ascertainment was used to make a FASD diagnosis by triangulating data from infant dysmorphology examinations, neurodevelopmental assessments, and maternal interviews. Microbial DNA was extracted from the stool samples and subjected to paired-end sequencing of the V1 to V2 region of the 16S ribosomal RNA gene. The DATA2 pipeline was used to pre-process the FASTQ sequencing files, construct an amplicon sequence variant table, and to assign taxonomy. Phyloseq, Vegan, and Maslin-2 were used to perform downstream analyses. Of the most abundant genera present in the gut microbiome, Faecilibacterium and Prevotella abundance was found to decrease from 6 weeks to 9 months of age. Infants with higher levels of Faecilibacterium tend to have better overall health outcomes, including lower rates of infection, reduced risk of allergies and asthma, as well as improved cognitive development. The decrease in the relative abundance of Faecilibacterium observed in this cohort is therefore concerning. Low abundance of Prevotella has been associated with increased morbidity as well as growth stunting, a common feature in FASD. Since the decrease in Prevotella abundance was observed in the FASD group only, this decrease may contribute to the growth and neurocognitive deficits characteristic of FASD. Interestingly, this is not the first time that maternal diet and nutrient intake has been highlighted in relation to FASD development in the same woman. Both of the highlighted studies show that women who drink large quantities of alcohol may be at an additional nutritional disadvantage, as chronic alcohol use can cause malabsorption or altered metabolism of nutrients. Therefore, maternal dietary intake during pregnancy may be an important FASD risk factor in which low BMI and nutritional status of the mother may exacerbate the effect of alcohol on the child. Although still exploratory, our study identified a potential microbial signature indicative of FASD development. Although further studies are required, these findings are promising for potential microbe-based therapeutic interventions which may reduce the extent of neurocognitive deficits and the debilitating symptoms associated with FASD. I would like to thank the following people and institutions. Thank you.
Thank you very much, Natasha. Really great presentation. I really was intrigued with the microbiome gut brain, and it's somewhere an area I'm studying when you're looking at this link, including the contribution of the oral microbiome and oral diseases to poor brain development. Looking forward to the networking session so that we can take this forward. We have one last presentation. Before I do that, I want to apologize for the interruption. I think we were hacked, but we have gotten the person out. Completely apologetic. So let's listen to Dyla now. Dyla Magini, who will be presenting the AWI Gene 2 microbiome project. I assume this must also come from the same lab as Samantha. Over to you.
Good afternoon, everyone. My name is Dylan McGinney, and I'm a postdoctoral fellow that works jointly between Stanford University in the US and the Sydney Brenner Institute for Molecular Bioscience in Johannesburg, South Africa. Today, I'll be introducing the AWIGEN 2 microbiome project. So I'm very privileged to be speaking on behalf of the AWIGEN 2 collective and specifically the AWIGEN 2 microbiome team, who are pictured here. This team represents a long-standing collaboration between Stanford and WITS and includes both postdocs and faculty from both institutions. So to begin, African populations are hugely underrepresented in microbiome research. This is perhaps best demonstrated by looking at the source populations for big microbiome reference collections like the Unified Human Gastrointestinal Genome Database pictured here on the left. And this database of microbial genomes from human guts around the world has strong representation from North America, Europe, and parts of Asia, with very minimal or no sampling from populations in South America, Africa, and Southeast Asia. This disparity represents a huge knowledge gap, especially as African populations have a distinct and shifting burden of disease pictured here on the right. Specifically, we see that African populations have a rising prevalence of non-communicable disease, but also a high prevalence of communicable disease, HIV, TB, malaria, etc. And as the gut microbiome is a known modulator of many of these diseases, it's imperative that we build comprehensive cross-sectional microbiome cohorts in African populations. So I'll introduce the AWIGEN 2 microbiome project. This project is part of the larger AWIGEN collective, which is a collaborative research effort to study genomic and environmental risk factors for disease in African populations. AWIGEN as a whole has enrolled 12,000 participants for genotyping and extensive clinical metadata collection. And AWIGEN places a strong emphasis on continual community engagement and genomic capacity building within the collaboration. AWIGEN operates in 6 study sites in Burkina Faso, Ghana, Kenya, and South Africa. As you can see here, these sites span a range of resource access and lifestyle, including rural areas, urban slums, and urban townships. Each site has a local research center that has been based in the community for decades, managing a full annual census of the study area and facilitating community engagement. So a huge advantage of operating within this network is that these long-standing community ties allow us to ensure that our project is addressing community needs and returning feedback to participants. However, the AWIGEN 2 microbiome project was not without its challenges. We were performing sample collection in the height of the COVID-19 pandemic, and field teams had to constantly adjust recruitment during national lockdowns and COVID spikes. Further, we had to consider cold chain and sample stability. We had a huge concern around batch effects and sample bias when shipping across the continent, and so we did a full partner study to evaluate stability of various stool preservatives, which you can find information on here. But all in all, AWIGEN 2 microbiome study recruited around 1,800 women from the 6 sites. This represents one of the largest shotgun sequencing gut microbiome studies on the continent and is paired with very comprehensive metadata, so we're very excited to dig into the analyses. At first glance, we can already see that these populations contain extensive bacterial diversity. So we assembled bacterial genomes from all the samples and put them into a tree, pictured here on the left. And we can see in the outer ring that we have genomes from all of the sites, spanning almost 20 phyla, which are colored here in the inner ring. Further, we can compare all of these genomes to existing genomes in reference databases like the UHGG, and we can see across sites that we have hundreds of genomes that don't match species in the UHGG database, implying substantial microbial novelty that hasn't been previously described in other studies. So in conclusion, I've shared the initial details of the AWIGEN 2 microbiome project. It represents a huge microbiome collection from 1,800 women across 4 countries. Critically, this research was done using existing study platforms that prioritize equitable research between collaborators and thoughtful community engagement. And this is only the start of the project. We have extensive clinical and environmental metadata to dig into. So hopefully what you've seen here is just the start of many projects seeking to understand that relationship between the gut microbiome and human health in African populations. With that, I need to thank the huge team that went into the microbiome project. There were so many field workers, site managers, biobank staff, and researchers across the study sites. So I need to thank everyone for their dedication and hard work that made this study possible. With that, thank you for your attention, and I look forward to discussing this further with you. Thank you.
Really great presentation. Once again, apologies for the consistent interruptions. We have about 5 more minutes leading to the top of the hour for the session. Please, if you do have any question you'd like to ask, exclusive of us in the chat box, please do raise your hands. And please let me know who you want to direct the question to. Remember, Professor Coker is still here for any question, and we have the 6 slide presenters. Any question from anyone around? Let me ask you, Professor Coker, while I wait for any question. And when I look at the microbiome gut-brain axis, and you definitely work in pediatric dentistry, and you also work in early childhood caries, is there anything you are trying to investigate where we're exploring the impact of early childhood caries through the microbiome on brain development? Is there anything we're looking in that direction?
Yeah.
Thank you.
Yeah, thank you, Prof. I think I love one of the earlier talks about the gut-brain axis, and definitely, the neurocognition, as you know, more than anybody, about the HIV, the cognitive impairments that come with HIV. Yeah. So one of the new projects that I have, which Mark,
apologies. Yes, Mark really wants it. Yeah. I'm sure we'll get Mark out now. Sorry.
Yeah, we have to get him out. So I think the answer to your question is we don't know enough yet. The gut-brain axis is definitely a lot of work. There's a lot of work about the astrocytes being impeded by gut microbiome translocation. So there's an interaction between astrocytes and gut bacteria. But for the oral microbiome, no. And more importantly, like my point of my whole talk, more importantly, nothing has been done with African population, which in my mind, you can't find a population of HIV-affected individuals more than in any other country. You can't find them beyond Africa, India, Africa, and that's it. So if you keep studying this in Western world, you won't have the large sample size that we require to see differences. Yeah.
Thank you. Thank you. I think Natasha has a question from the chat box by Johanna. It says, how early in infancy do this gut microbiome changes become evident in individuals with FASD? And do they persist into childhood or beyond? That question goes to Natasha.
Thanks so much for the question. So it's a really good question. Unfortunately, we're not quite sure. Our study only looked at 6 weeks and 9 months of age. So we're not quite sure at what point between that 6 weeks and 9 months these changes occurred. But we are doing a follow-up study, which includes more time points, including up until 5 years of age, so that we can pinpoint sort of what time period these changes occur, and therefore hopefully able to at least do something to try and mitigate some of the effects.
Thank you. You have one more question before you leave. It says the current study investigates the microbial differences between rural and urban cohorts. It cannot show how quickly. Oh, geez, that went off so fast.
Oh, I think that's Matsepo's response to me. Not a question, but thank you.
All right, thank you. Dyla, I think we'll take the last question. Dylan, apologies if I'm messing up your name. Please forgive me. It says, thank you. It's a huge cohort with extensive clinical and demographic data, so we're very excited to dig further into the analysis and collaborate when the data is deposited. Or I think that was a response. Otherwise, I'm not sure we have any other question. We just have a minute to go. Otherwise, we'll move into the next session. Awesome presentation, everyone. Really educative. Looking forward to the networking space and engaging with you with further discussion. Thank you. So we'll move on to the next talk. And this is coming all the way from Tunisia. Dr. Sumaya Koinde, who will be discussing with us this afternoon, evening, morning about elevating health precision, unveiling microbiome and metabolomic impact in breast cancer, kidney disease, and stem cell therapy. Sumana is from University of Manouba in Tunisia and works on microbiome, drug resistance, and cancer. Specifically, she has successfully incorporated innovative methods to explore the gut microbiome and metabolome, which she has applied in the field of organ transplantation and oncology. So it's a pleasure to have you here and looking forward to hearing from you over the next 15 minutes. Thank you. Over.
Thank you. Can I just share my presentation? I'm not able to share my slides.
Presentation. Please, could you help enable Jumania to be able to share slides, please?
That we're trying to contain the situation, so we've kind of disabled the right now. So yeah.
Okay.
Excellent. Thank you.
Can you see my slides?
Yes, please. We have it. It's gone now. Would you share again, please?
Okay, so first I would like to extend my heartfelt gratitude to the organizing committee for entrusting me with the privilege of being part of the second African Microbiome Day. A warm welcome to all the attendees who have joined us today. Our collective journey promises...
You have to share the slides again. We lost it. Yeah. Excellent. It's coming up now. Yes.
Okay.
Then the full slides.
Is it okay?
Thank you. We have it now. Thank you.
Okay. So let's begin this exploration of metabolic and microbial frontiers, where we aim to shape a healthier future for all. Certainly, the microbiota is a matter of crucial importance for the future of medicine, but the topic was addressed 2,400 years ago by Hippocrates, who proclaimed that all diseases begin in the gut. And the science is increasingly confirming this theory with the advent of high-throughput sequencing and the Human Microbiome Project launched in 2007. The last decade has been marked by a significant rise of in data highlighting revelation of the essential role of microbiota in health and pathologies. The human microbiota, also known as the hidden organ, contribute over 140 times more genetic information than that the entire human genome. The composition of microbiota, as you can see, varies from site to site, but the gut microbiota is considered the most significant one in maintaining our health. The gut microbiota is crucial to shape host physiology and interact with the host in different ways. It acts as a commensal metabolic organ. It can regulate our immune system and prevent pathogenic bacteria and viruses from colonizing our gut. However, the microbiota can undergo alterations due to the exposure to various environment factors, diet and drug we take, such as antibiotics or intense treatment like chemotherapy, which result in changes in taxonomic composition and associated functions. This is referred to as dysbiosis, which occurs when there is a reduced bacterial diversity, a loss of beneficial bacteria, or an overgrowth of pro-inflammatory invasive bacteria. So if the balance tilts and pathogenic microorganisms gain the upper hand, this significantly increases the risk of illness such as inflammatory bowel diseases, metabolic disorders, neurodegenerative diseases, and cancer. So it is not surprising that in the 14th Cleveland Clinic's Medical Innovation Summit, which is the oldest and the largest meeting of its kind in healthcare, clinic experts predict that among the top 10 medical advancements, using microbiome to prevent, diagnose, and treat disease will change patient care forever. In the same year, I mean in 2016, President Obama made an announcement about initiating the Precision Medicine Initiative. One of its crucial strategic objectives was to emphasize the necessity of integrating diverse patient data with a specific focus on metabolomics and microbiome. Furthermore, in precision medicine, the utilization of multi-omics approach is becoming more prevalent, especially in microbiota investigations. A noteworthy example is the Integrative Human Microbiome project, which was initiated in 2012, which placed significant emphasis on integrating various omics data, such as metabolomics and metagenomics. The results underscore a strong connectivity between microbes and metabolites in health and disease. This relationship is particularly intuitive since the microbiota and the metabolome are influenced by shared upstream factors. So, better understanding the close relationship between the metabolome and the microbiota in disease is therefore an important and emerging research objective. Given the lack of precise diagnosis, the heterogeneity in therapy response, and consequent development of complications, particularly in kidney graft, in hematopoietic stem cells, and in breast cancer, we have set the main objective as so the implementation and optimization of new methods and the identification of new invasive biomarkers, I mean bacterial and metabolic biomarkers. Thus, we have set out to conduct a targeted study, metagenomic study 16S, and a metabolomic analysis of gut microbiota applied to organ transplantation and breast cancer. Our first study model is kidney transplantation. Recent studies demonstrate the existence of relationship between the kidney and the gut microbiota, referred to as the gut-kidney axis. Dysbiosis is particularly critical in kidney transplant recipients, given the risk of graft rejection and increased susceptibility to infections. But despite the significance, the clinical measures used for monitoring these patients remain relatively simplistic and lack sensitivity and specificity. Hence, the urgent need for early, more robust and less invasive biomarkers. So, our results of the metataxonomic analysis revealed significant change in the transplant group, which exhibited lower bacterial diversity and richness, marked by a decrease in beneficial actinobacteria and a gradual increase in proteobacteria, which is a phylum often associated with inflammatory disease. At the genus level, there was a notable decline in beneficial genera that produce short chain fatty acids like Faecalibacterium and Prevotella, accompanied by an increase in pro-inflammatory pathogens, particularly in patients with associated diseases. These microbial shifts have been linked to a renal insufficiency. Then, the co-occurrence analysis showed fewer bacteria interactions in renal transplant recipients compared to the control group. And proteobacteria had high interactions, inhibitions rates in the transplant group, especially in those with associated diseases. Further, the genera Succinio vibrio and Suterella displayed mutual exclusivity in this group, possibly tied to dysbiosis-related complications. At the metabolic level, the fecal metabolome in kidney transplant patients significantly differ from the control group. We identified 18 key fecal metabolites indicating increased fecal lipids and decreased vitamins and microbiota beneficial metabolites. Several pathways include ubiquinone and terpenoid quinone biosynthesis, tyrosine metabolism, tryptophan biosynthesis, and primary bile acid biosynthesis appear to be altered and correlated with renal dysfunction or nephrotoxicity. Our second model is hematopoietic stem cell transplantation. Allogenic hematopoietic stem cell transplantation represents the most effective and well-established form of immunotherapy of various hematological malignancy. However, it is hindered by high mortality and morbidity rates attributed to several complications, including bloodstream infection and graft versus host disease called GVHD. Today, early diagnosis tests and predictive biomarkers for these complications or therapeutic response remain quite limited. Our metataxonomic analysis revealed lower bacterial diversity in allograft group, aligning with previous studies. Phylum level differences included a decrease in firmicutes and bacteroidetes and a notable 30% increase in proteobacteria in patients compared to healthy controls. At the genus level, allograft recipients displayed altered microbiota composition with a distinct profile in those developing GVHD. This profile featured reduced beneficial genera and increased pro-inflammatory pathogens. Further, the co-occurrence analysis of bacterial interaction showed significant changes at phylum and genus level. Proteobacteria had high inhibition rates in the allograft group, and in the GVHD group, two potentially pathogenic genera, Bacteroides and Citrobacter, exhibited numerous mutually exclusive interactions, possibly related to dysbiosis-related complications. At the metabolic level, the metabolic analysis revealed distinct fecal metabolome signature in patients compared to control group, with 24 identified metabolites, notably patients exhibited elevated level of oncometabolites, fumaric acids, and succinides, alongside decreased microbiota beneficial metabolites and vitamins. Altered pathways include tricarboxylic acid cycle, amino acid, pyruvate, and glycolysis metabolism. Moreover, pathways associated with acute GVHD, especially related to the intestinal microbiota and bile acids, displayed significant change. These findings align with existing literature and correlate with metabolic reprogramming, immune alterations, and GVHD occurrence. Our third study model is breast cancer, which is the most common cancer and the second leading cause of death among women worldwide. In Tunisia, it represents 34% of female cancers. Managing this disease poses a significant challenge to the major medical issues associated with treatment protocols. Hence, the need for biomarkers for prognosis and response to anti-cancer treatments. Recent studies have emphasized that the microbiota is an important emerging biomarker in cancer. The microbiota can modulate both the tumor and the tumor microenvironment. It is potentially an instrumental and quasi-independent variable in the puzzle of how cancer develop, progress, and respond to treatments. Our results showed that at the metataxonomic level, there is a different composition of the microbiota over the course of the three cycles of chemotherapy. It is interesting to note that as the cycles progress, there is an increase in beneficial bacteria described as biomarker of good health, such as Bifidobacterium and Faecalibacterium primary in good responders. The metabolic analysis showed distinct metabolic profile shift post-cycle 2 and cycle 3 of neoadjuvant chemotherapy. We identified 27K fecal metabolites that differentiated the groups effectively. This finding included reduced oncometabolites, lactates, fumaric acid, and succinate, and all Krebs cycles intermediates, and increased amino acids known as serum urine and plasma biomarkers, alongside specific microbiota beneficial metabolites such as short-chain fatty acids linked to enhanced treatment efficacy with higher level. To refine the biological explanation, we identified alteration in metabolic pathways, including amino acid metabolism, glycolysis, pyruvate metabolism, tricarboxylic acid cycle, tyrosine metabolism. These metabolic findings were corroborated by existing literature and linked with metabolic reprogrammation and cancer cell surviving and proliferation. The overall results are quite promising for an interdisciplinary approach and participatory research, an implementation of innovative approaches for specificities in the structure of intestinal and metabolic feature inherited to different studies pathology, accurate discrimination between patients and controls, for improved understanding and characterization of human physiology, enable progress toward personalized medicine. All of our results have led to publication in peer-reviewed journals. In perspective and taking into account the main application of the microbiome in medicine, let us recall that microbiota use include biomarkers where patients are screened, monitored, and stratified, and the therapeutic use where there are currently 9 forms of therapeutic dietary intervention: prebiotic, probiotics, symbiotics, antibiotics, fecal microbiota transplantations, phage therapy, live biotherapeutics, and the end generation, the microbiome mimetics. For the first use, and in light of our findings and our local context, it is possible to develop more targeted, faster, and cost-effective approaches for the molecular and functional characterization of the microbiota to enhance patients' monitoring. This includes creating a set of validated metabolic and bacterial biomarkers and adopting targeted metabolomic and qPCR techniques stemming for the genuine necessity and in-depth consultations with collaborating clinicians. For the second use, in our microbiota-centered therapeutic approach, we've initiated a bioinformatics investigation into the inflammatory mechanism influenced by the microbiota, particularly focusing on the screening of the molecule regulating TLR2 and TLR4 receptor. This novel approach has identified the top 10 bioactive molecules produced by the microbiota that exhibited significant interaction with these crucial immune receptors. Currently, functional tests are underway in animal models to evaluate the impact of this molecule on inflammatory response. We eagerly anticipate presenting the results of the next event, signifying and significant advancement in microbiota-based therapy. So as a key takeaway, microbiome-based medicine have advanced dramatically over the last decades. The primary challenge now faced is in identifying clinical diseases amenable to intervention with microbiome-based medicine. And microbiota targeting and management may provide a more effective, sustainable healthcare in Africa. Because the majority of diseases begin in the digestive tract when good bacteria are no more able to control bad bacteria. So I would like to thank all my collaborators and my partners that who have contributed to the completion of this work. And thank you for your attention.
Thank you, Dr. Kwendi. Excellent presentation, excellent timing. And yes, I leave away that microbiome is the future of medicine and dentistry. And that personal care and microbiome studies becoming, a key for disease solutions. The many diseases we still don't have solutions about. Thank you so much. The space is open for questions. We've come to the end, but if you just have one question, we're just on top of the hour. But if you do have one critical question, thank you. There's a question from Professor Shitu who says, interesting talk. I was just thinking of asking you about the role of nutrition in cancer treatment, prebiotics, probiotics, postbiotics. What are those roles in cancer treatment? Thank you. Did you get the question, Dr. Kwendi? Dr. Kwendi, are you there?
Sorry, can you repeat the question? I didn't hear you very well.
Okay. So what's the role of nutrition in cancer treatment in terms of prebiotics, probiotics, and postbiotics? What are the roles of this in cancer treatment?
Of nutrition?
Yes. Prebiotics, probiotics, and postbiotics.
Yes. There is a solid data in favor of good nutrition. Today, it is well known, for example, for the immunotherapy treatment for cancer. It is well known today that a good nutrition, especially there is an interesting work comparing the Mediterranean nutrition and the Occidental nutrition, and they show that this kind of nutrition can improve significantly the response to immunotherapy. So there is a lot of work now that argue this direction. So today, I think that that clinicians and nutritionists will talk about preventive medicine through nutrition, through good nutrition, even through prebiotic and probiotic to have a good response to treatment of cancer. So there is a lot of data in this sense.
Thank you so much. Thank you, really. I thank you for everyone for participating in this session. I've gone over a session by 3 minutes. I sincerely apologize. I'd like to hand over to the next session and thank all our presenters, Professor Kolka, Professor Kwendi, the 6th abstract presenter. It was stimulating and the best way to have started this conference. Thank you, everyone. Continue to enjoy the session, and over to you. Over to the next session. Thank you.
Okay. Thank you very much, Professor Muraki, for the first section. I hope you can all hear me clearly. Right?
Yes, we can. Very loud and clear.
Okay, thank you very much. My name is Dr. Lamek from Namibia University of Science and Technology, based here in Namibia. I'll be taking you through the second session of reproduction and breast milk microbiome. Maybe before I begin, I'd like to mention that we apologize for the interruptions that have taken place, and we have really taken measures to contain them, and hopefully this won't happen again. Right, so to just take us through to this second session, we're going to have three speakers. 2 invited speaker and 1 poster section. So our first invited speaker is Professor Joanne Passmore. Professor Passmore's lab focuses on studying the genetical, tract adaptive, and innate immune responses associated with protection from or susceptibility to sexually transmitted diseases. This includes HPV and HIV. Specifically, Professor Passmore's lab is interested in studying biomarkers for susceptibility and protection to HIV infection, including secreted inflammatory molecules, genital immune activation, and HIV-specific immune responses present in the female genital tract. Professor Passmore, your time starts now. Thank you.
Thank you very much for the invitation. And I would like permission to share, if possible. There. Thank you very much. So I'm representing a large number of people. So I'm very grateful to have the opportunity to talk on their behalf, but to capture some of the work we've been doing in South Africa with partners in Kenya in establishing the Vaginal Microbiome Research Consortium for Africa, and that's our website. So I'm going to tell you more about how we got to this project than results from the study, because the clinical studies have started in South Africa and Kenya relatively recently, and hopefully I'll be invited back in a couple of years' time, where we'll have some really rich data to share and discussions for collaboration. So I am based at the University of Cape Town, in Cape Town, and I'm very aware of microbiome research across the continent. And in particular, a couple of studies stood out to me, which were really just taking stock of publicly available microbiome data and which countries were publishing it. Because during my previous life, I participated in and benefited from H3Africa had a program called H3Bionet, which trains significant number of computational biologists, bioinformaticians around microbiome pipelines. And that program is now come to it's shrunk, but it's left this imprint of a substantial amount of microbiome data across the continent. So I've presented this in the past, and I think this is from a PLOS biology paper that came out last year, where they had tracked the number of samples that were publicly available. And as you will see in the pink stack bar, a significant proportion of this comes from Europe and North America, and less comes from the region I'm representing, Sub-Saharan Africa, in the orange. And although the size of the stacks per year is growing substantially, it's growing disproportionately in Europe and North America. And obviously this reflects really strategic investments from the NIH and European funding agencies to have a roadmap for microbiome research that they started more than 10 years ago. And that's, it's that major sort of driving force that included funding, but also high-level discussion on how to incentivize this kind of research. So this paper came out from people that are part of this setting up this workshop, this symposium today, but others who participated in H3Bionet. And it was a systematic review of the microbiome research across Africa, looking at the different sites from which samples were being obtained, the countries that had deposited this sequence. And actually, although the previous slide looked quite dismal in terms of how much publicly available data is deposited from Africa, in particular sub-Saharan Africa, there really is quite a lot of microbiome research going on across the continent. But the downside of this is when they tracked publications coming out of this publicly available data, reflecting the funding channel from the west to the south, or the north to the south, a significant proportion of papers had first author affiliations with the USA as well as last author. So the publications, when they were published, come from senior authors or lead authors who were in the global north, and less so for researchers in Africa. So what we really sort of took stock of from this was how to shift this to an African-led agenda as more data comes aboard, to try and bring together this significant amount of know-how and expertise that exists on the continent, to have a more strategic vision of the microbiome landscape, and to harness that leadership potential from the investments for training that have happened on the continent. So the conceptualization, and this wasn't my brainchild, it's a cohesion of really important collaborations I've had over the years, but to work with South African and African partners that have the capacity to do regionally relevant vaginal microbiome studies, and importantly contributes, because I work in a space of bacterial vaginosis and HIV risk, which I'll tell you about in the next couple of slides, to contribute vaginal lactobacilli, including genome sequences and functional characterization from women living in either South Africa, Kenya, or the countries with which we are collaborating. And then to use this information about the microbiome, stability over time, as well as properties of the vaginal lactobacilli, which we as a team isolate and characterize, to develop novel therapeutics for treating common dysbioses like bacterial vaginosis. And I'm going to tell you a little bit about how we got here and why this is really important if you're a reproductive tract scientist working in particular HIV prevention, which is how we got there. So for 20 years at the University of Cape Town, together with Caprisa in Durban, Desmond Tutu HIV Health Foundation, the core focus, because of the unacceptably high burden of HIV risk in women, in particular young women, a lot of the research we've been doing has focused on understanding HIV risk and preventing HIV infection in women. And our lab has gone about this two ways. There's a lot of asymptomatic infections on the continent and in South Africa, and we've been developing a screening tool, a genital inflammation test to screen for asymptomatic infections that can be treated efficiently with antibiotics. But then conditions like bacterial vaginosis is a dysbiosis. It's not a sexually transmitted infection. It isn't perfectly or even nearly perfectly treated with antibiotics like metronidazole. So finding ways to improve reproductive health using live biotherapeutics based on vaginal lactobacilli from women who really have a robust and stable vaginal microbiome. So this work started with a really important collaboration with Caprisa in Durban, and women were enrolled from around Durban city, so urban, as well as from Vulindlela, which is a rural site just outside Pietermaritzburg. And this work is relatively old now, published in 2016, showing that genital inflammation that exists prior to HIV infection increases a woman's risk for acquiring that HIV infection. And as a mucosal immunologist, spend most of my time focusing on cytokines in cervicovaginal fluid, and in particular these MIPs, macrophage inflammatory protein 1 alpha and 1 beta, and RP10 and interleukin 8. And if we stratified in a case control study women who went on to seroconvert and become HIV positive, but this is prior to infection, versus women who remain negative during 2 years of follow-up, that significantly more women in the seroconverter case group had elevated MIP1 beta, MIP1 alpha, RP10, and IL-8, and in a matched pair odds ratio, having any of these cytokines, the MIPs elevated, were associated with an odds ratio of 3, meaning that these women, just by having those chemokines present, they were at a higher risk for HIV infection. So when we binarize this into genital tract inflammation being present or absent, and we defined inflammation based on having more than half of the pro-inflammatory cytokines and chemokines being in the 75th percentile of the cohort, that the odds ratio in this binarized way for HIV infection was 3.2. We went on to look at what the major drivers of inflammation were in this cohort, because if you could understand the cause of the inflammation, you could intervene. And we looked at treatable STIs first, and none of the treatable STIs, including T. vaginalis, chlamydia, and gonorrhea, perfectly explained inflammation. So only 20% of these HIV infections were attributed to any of these classical STIs or treatable STIs, with T. vaginalis being the most strongly predictive. In the study at the time it was done, we hadn't conducted BV scoring, Nugent scoring, and no microbiome studies were conducted. So we've spent the last 10 years really interrogating the vaginal microbiome to look at the causal relationship with genital inflammation. So this work hasn't been published, but it was presented at the AIDS conference in Durban in 2016. And it was a collaboration between us at Caprisa and Ian Lipkin and Brent Williams at Columbia. And these are the same cases and controls I showed you in the previous slides. But here, the little red bars are the seroconverters, subsequent seroconverters, compared to the gray bars, which are women who remained HIV negative. And then looking at the cytokine groups, this orange is women with elevated inflammation, and green, irrespective of the color green, both greens were women with low levels of inflammation. And so this analysis basically shows concordance between HIV infection and cytokine status, and the microbiome of the majority of women that had Lactobacillus crispatus also had low levels of inflammation and remained HIV uninfected down here. So the community state type 1 was a classification that was used, which is dominated by Lactobacillus crispatus. The majority of the cohort—
You have 1 minute, eh?
Okay, thank you. Had CST3, which was inners, and really a mixed phenotype. but the majority of the HIV infections and inflammation were explained by Prevotella, Bivia, and Snedia. And Prevotella bivia, which is a common component of bacterial vaginosis, was the best predictor of genital inflammation and HIV risk in the cohort. We corroborated this, and this work is elegantly led by Heather Jaspin at the University of Cape Town and Katie Leonard, where in a completely independent cohort of young women in Cape Town, we showed that being bacterial vaginosis positive was a strong predictor of inflammation, and Prevotella bivia in particular was the strongest driver of that inflammation. So going back to Vaginal Microbiome Research Consortium, and what the idea behind this was to develop, to study lactobacilli that could be used as a novel therapeutic for treating bacterial vaginosis, because metronidazole does such a terrible job, and in addition conduct testing of these vaginal products. And some of these trials are starting as we speak at Caprisa, with parallel studies being done in Boston. Phase 1 was leveraging samples we already had in our freezers, so biobanked specimens from longitudinal cohorts that Heather and I had funded through the NIH that enrolled women in Cape Town. And this work has been led by Brian Cullen, and it was part of Annika Chicken's and Kirsten Welp's masters, which they're almost finishing. So we've actually screened 337 lactobacilli isolates, and we're looking for the ability to inhibit Prevotella bivia, and we're finding really good, exciting ability to inhibit this really inflammatory Prevotella bivia, and these are all L. crispatus isolates. So basically the pipeline's full of strains of lactobacilli from these convenient samples. And then ending off here, we have secured funding through the Gates Foundation to pilot Vaginal Microbiome Research Consortium for Africa, with 2 pilot sites, one through a really good collaboration with the Desmond Tutu Health Foundation, and with Sarah Gatomi and Prof. Elizabeth Bakuzi in Kemri. And the study design for that is a 10-week intensive sampling, 2 samples per week, self-samples by the woman. And we are collecting speculum exam samples at 3 time points, but it will really give rich data on the transition states over 2 menstrual cycles in women from 2 African countries. And we're enrolling currently 100 women from Cape Town and 100 women through Kenya, through Kemri, and lots of samples being stored and selected. And then I just really wanted to just give a shout out to others on the call from Africa. Just, never underestimate the really core value of doing research on the continent, and not opting to go the often quicker routes, cheaper, where samples get shipped elsewhere, or to work out some equitable partnership where there's really investment in skill development, because Africa has a strategic advantage in that we've got a population in need. We've got amazing centers of excellence for recruiting cohorts, taking samples. We've established significant biobanking capacity on the continent, and it's sort of lower down this pyramids of skill that I'm really pushing and urging us to negotiate better, and that would be to do the sequencing and the bacterial strain isolation in the countries in which the samples are collected, but also to be really key partners in the translational sciences. So I'm going to end off with a really long list of acknowledgements and to acknowledge funding from the Bill and Melinda Gates Foundation through the Colesta Schumer Scientific Leadership Fellowship to our team. And our team is a very long one. So these are the people I'm representing on the call today. Thanks very much.
Thank you very much, Prof. I'm very glad at what you've just spoken about in terms, because this event is all about if we can be able to network. And from your slides, initially they actually gave a very grim picture of the fact that there's not much research being done in Africa, or the research being done a lot in Africa is you find that you have a lot of these either first authors or senior authors coming from different regions. So it's a good food for thought. This is something that we should actually be able to discuss during the breakout sessions. Right, so I will not be taking questions now, but I hope that anyone who has questions, please write them in the chat box. And then after the third presenter, then we can have all these questions together. So I'll now take us straight to Lauren Martin, the poster presentation. Please, Lauren.
Hi everyone, my name is Lauren Martin and I am a PhD student in the Neuropsychiatric Genetics Research Group at Stellenbosch University. And today I'm going to be telling you a little bit more about my master's work, with particular focus on the iSeq 100 study that was embedded within it. So we have previously investigated the maternal vaginal bacterium in relation to fetal alcohol spectrum disorder development, and we used vaginal swab samples collected from 28 women, as well as data collected from their infants. Unfortunately, because of the sample size, no definitive conclusions could be made. And furthermore, we were only able to confidently discriminate bacterial taxa to genus level, as we'd made use of targeted V1, V2 hypervariable sequencing data. However, the small sample size did present us with a very unique opportunity in that we were able to inexpensively resequence these samples on different platforms and perform some comparisons. In fact, the sample set was particularly useful when we were approached by Illumina and asked if we'd undertake a pilot study to validate the iSeq 100's ability to sequence the full-length 16S rRNA gene in 150 base pair-end reads. And this is quite important to establish because the platform has lost a lot of functionality through its inability to sequence several hypervariable regions, and this is largely due to the read length constraints. So bearing that in mind, we aim to improve the species level resolution of the bacterial communities present in the maternal vaginal swab samples collected from 28 alcohol consuming and abstemious women who gave birth to infants with and without FASD. And we did this through performing long and short read sequencing of the full-length 16S rRNA gene using the PacBio Sequel 2E instrument, as well as the Illumina iSeq 100. Thereafter, we evaluated the extent to which data characterizing the full-length 16S rRNA gene outperformed that of the targeted hypervariable sequencing data in terms of taxonomic classification to species level. Following library preparation and sequencing on the iSeq 100, we then needed to assemble those reads into the full-length 16S rRNA gene. Unfortunately, many of the programs that are capable of doing this were outdated or had their support languished by the developers. So with the invaluable assistance of Dr. David Twesi-Gomwe, we developed an easy-to-use Nextflow pipeline, which we called 16S AmpliconSeq, that performs gene assembly, taxonomic classification, and then also further facilitates microbiome-related processing and analysis in R. Just in terms of results, the PacBio and the iSeq 100 datasets had comparable species-level taxonomic performance, with the PacBio dataset outperforming the iSeq 100 dataset by a mere 3%, providing evidence of the feasibility of our sequencing technique and our bioinformatics pipeline for use in future studies. And with that, I'd like to acknowledge all the people involved in the study, as well as our funders. And then lastly, thank you for listening.
Okay, thank you very much, Lauren, for that presentation. I think I'll take us straight to the second invited speaker of this presentation of this section, and this second invited speaker is Dr. Anna Happel. And as I said before, we are going to take Q&A questions before we go to the break. So, Dr. Happel, our current work involves understanding interactions between bacterial and viral microbiota in the female genital tract and subsequent effects on risk in preterm birth in women with HIV. And she ultimately aims to use her research to design microbiome-based diagnostics and biotherapeutics for women's and infant health. So today, Dr. Happel will be taking us through a talk titled The Impact of In Utero HIV Exposure, Feeding and Maternal Breast Milk Microbiota and on Infant Gut Microbiota and Early Childhood Vaccine Responses.
Yeah,
Dr.
thanks
Happel,
so much.
take it on. Yeah.
Thanks so much for the kind introduction, and I'm excited to present some work that I've conducted during my time as a postdoctoral fellow at the University of Cape Town under Dr. Heather Jaspin's mentorship. So as a brief introduction, over the past 20 years, the number of pregnant women with HIV has remained stable. What has, however, changed is the global coverage of antiretroviral therapy amongst pregnant women living with HIV. With moving to the area option B+, all individuals with HIV, independently of their CD4 count or viral load, are encouraged to receive antiretroviral therapy, and this includes pregnant women. This has led to a decrease in the infants acquiring HIV perinatally, as shown in the blue line. What this, however, means is that the global population of infants who are HIV exposed but uninfected is substantial and continues to grow. So these are infants who are born to mothers with HIV, but they are not HIV infected themselves. And in South Africa, due to the high prevalence of HIV, about 1 in 4 children are exposed to HIV but uninfected. This population of children is vulnerable because they're at an increased risk for infectious and non-infectious morbidity. This includes skin, mucous membrane, and respiratory tract infections, a high incidence of group B streptococcus infection, and suboptimal growth. The causes for this increased risk of morbidity amongst infants born to mothers with HIV compared to infants who are born to mothers without HIV are likely to be multifactorial. And it is hypothesized that microbiome alterations might play a role because previous studies have shown that HIV-exposed infants have a lower diversity in their gut and also, generally an altered gut microbiome composition. Infants who are exposed to HIV also show immune alterations. This includes lower CD4 T cell counts, elevated immune activation, and altered vaccine responses. More recently, there's some data emerging that the human gut microbiota might influence the immune development. For example, it's been described that Bifidobacterium infantis, which is one of the earliest colonizers of the infant gut, induces regulatory T cells. In mouse models, it's been shown that filamentous bacteria can induce inflammatory Th17 cells. It's been shown that peptidoglycans from gram-negative bacteria can induce the recruitment of B cells to the gut, and some lactobacillus species induce the maturation of dendritic cells. Lots of those studies have been conducted in vitro or in mouse models, but there's some, like human data, emergent from observational studies that describe an association between infant gut microbiota and vaccine immunogenicity. This includes studies that have described an increased abundance of bifidobacteria and its association with beta BCG, tetanus, and hepatitis B vaccine responses at 15 weeks of life. Regarding rotavirus vaccine responses, the data is, or the outcomes of the studies are relatively mixed. Some studies have described other associations while others haven't. And there are few data on other early childhood vaccines, including pertussis vaccine. And pertussis vaccine is an important vaccine for infants who are HIV exposed and uninfected, since they're at an increased risk of respiratory and mucosal infections. So just briefly summing up the rationale for the study, breastfeeding is recommended by the World Health Organization for women with HIV, thus infants who are HIV but exposed but uninfected are a key population for HIV vaccination. And it's been described that HIV-exposed infants have an altered immunity, including altered T-cell responses to vaccination. So what were our two main questions that we wanted to answer with this project? One of them was whether the maternal breast milk microbiota and infant gut microbiota differ in a South African cohort by HIV exposure stages, despite the increasing use of maternal antiretroviral therapy during pregnancy, because many studies that previously have described differences in gut microbiota by HIV exposure stages were done during a time where universal antiretroviral therapy was not recommended. And the second question was how the infant gut microbiota relates to pertussis vaccine responses. We hypothesized that altered maternal breast milk microbiota contributes to altered gut microbiota in the infants and thereby contributing to altered pertussis vaccine responses. Heather Jaspin's clinical site is based in Khayelitsha in Cape Town. It's a peri-urban settlement with a high prevalence of HIV. About 30% of the women who we approach in the midwife obstetric unit for enrollment have HIV. And for the study, we limit enrollment to women who delivered their infants vaginally. Enrollment happens at birth, where mothers and their infants are enrolled. We follow them over 9 months with regular follow-up visits, where both extensive metadata as well as a bunch of samples is being collected. We collect infant and maternal stool, as well as maternal breast milk for microbiome studies. And we collect infant blood to measure vaccine responses, but also to conduct HIV DNA PCRs to confirm that the infants who are HIV exposed but uninfected really remain HIV uninfected and do not acquire HIV postnatally. And for the data I'm going to present just now, we've included 50 mothers with HIV and their HIV exposed but uninfected infants, and 25 mothers without HIV and their HIV unexposed infants. We conducted 16S rRNA gene sequencing of the V3, V4 region of breast milk, shown in red, infant stool shown in green, and maternal stool shown in blue, collected at day 4 to 7 of life, weeks 4, week 15, and week 36 of life. And what you can see in the first plot here is that the samples clearly cluster by sample type as expected, since the bacterial composition of breast milk, infant stool, and maternal stool differs. I'm going to go into the compositional aspect a little bit later in more detail, but I just wanted to show here also that the diversity of the different sample types differ. So maternal stool, shown in blue, on the right-hand side had the highest diversity, while breast milk and infant stool had a lower alpha diversity. But as time passed by over the course of the study, the diversity of breast milk and infant stool increased and became more similar to that of maternal stool. Next, we looked at whether HIV infection has an effect on the breast milk microbiota composition. In the bar plot at the bottom, you can see that the breast milk microbiota was dominated by Streptococcus oralis, Streptococcus salivarius, and Staphylococcus caprei. Those are all either skin or oral bacteria, and this is in line with what has been described previously in the literature. When we look at the alpha diversity, we can see that there were no significant differences in the breast milk collected from mothers without HIV, shown in orange on the left-hand side, and mothers with HIV, shown in purple on the right-hand side at any of the time points. Similar, when we looked at beta diversity, we did not see any significant clustering by maternal HIV status at any of the time points, indicating that the overall composition of the breast milk microbiota did not differ substantially between women with and without HIV. And we conducted some DSeq2 analyses to see whether there were any differences in specific bacterial taxa at any of the time points. And across time points in breast milk collected from women with HIV, there was an increased abundance of Streptococcus pneumonia and a decreased abundance of Corynebacterium croupenstati, which has previously been associated with mastitis and Streptococcus parasanguinis. Next, I'm going to share some data on the gut microbiota of stool collected from HIV-exposed and unexposed infants. Similarly to the breast milk, we did not find any differences in alpha diversity in the stool microbiome from HIV-unexposed infants, shown in pink, and those from HIV-exposed infants, shown in yellow. When we looked at overall bacterial community composition, we saw that there was some clustering at week 4 of life, suggesting that the community composition differed at week 4 of life between HIV-exposed, again shown in yellow, and HIV-unexposed infants, shown in pink. I would like to draw your attention to the shape of the data points used in the plot, because the shape indicates whether an infant was still breastfed or was already completely weaned and did not receive any breast milk anymore at week 4 in life, but was rather formula fed. And you can see that all the pink data points are squares, indicating that all HIV-unexposed infants still received breast milk at week 4 of life, while there are some HIV-unexposed infants who did not receive any breast milk anymore at week 4 of life. And I mean, there's extensive literature that mode of feeding influences the gut microbiota. So in the subsequent analyses, I adjusted for mode of feeding. And when adjusting for mode of feeding, there were actually very few differentially abundant taxa in the gut microbiome of HIV-exposed infants compared to unexposed infants. At day 4 to 7 of life, HIV-exposed infants had an increased abundance of Bifidobacterium cantinulatum, and at week 15, they had increases in Delphtia and decreases in Haemophilus influenza. But other than that, we did not find any differences, which led us to conclude that the differences in gut microbiota composition at week 4 of life are largely driven by mode of feeding. I conducted some supplementary analyses looking at associations between mode of feeding and infant gut microbiota composition. And indeed, at week 4 and week 15 of life, the community composition differed by mode of feeding, and infants who still received breast milk at these time points tended to have a lower gut microbial alpha diversity. This is nothing new. There's been extensive literature that breastfeeding is beneficial and impacts infant gut microbiota, but it was just good to confirm that the findings, the differences that we see at week 4 of life between HIV-exposed and unexposed infants is indeed largely driven by mode of feeding. Then I spent quite a lot of time looking at the relatedness between maternal breast milk and infant gut microbiota. So this is the output of a network analysis where sample types are again indicated through the shape of the data point, and the colors indicate the participant ID. And mothers and infants in this plot have the same color. So you can, for example, see that this mother here donated 3 breast milk samples, and here we have a matching stool sample from her infant. And you can see that there was primarily clustering by sample type and not so much by mother-infant pairs. And if there were significant relatedness between samples, there's a line between data points. So on an overall community composition, we did not see strong relatedness between maternal breast milk and infant stool microbiota. Another way to look at this is using PCOAs by time point, where again, the shape indicates the sample type and the color indicates the participant ID. And you can again see that samples rather cluster by sample type than by mother-infant relatedness. I conducted some other analyses that I'm not going to share here now that looked at whether, rather than rather than looking at similarities between overall community composition, I looked at whether mother-infant pairs shared specific bacterial species or specific ASVs. But again, we did not see many similarities between maternal breast milk and their infants' gut microbiome composition. Next, I looked at associations between infant gut microbiota and pertussis vaccine responses. So pertussis vaccine is given in South Africa at 6, 10, and 14 weeks of life, and at 18 months there's a booster. And we measured pertussis vaccine responses using an ELISA. And there was a pre-specified threshold shown in the dotted line that determined whether an infant was a responder or a non-responder. And you can see that at birth and week 4, which were both time points prior to the first vaccination being given, we did not have any responders or few responders as expected. While at weeks 15 and 36 post-vaccination, some infants had sufficiently strong responses that were deemed that classified them as a responder, and the proportion of pertussis vaccine responders was higher amongst infants who are HIV exposed but uninfected. I next looked at associations between infant stool microbiota and at the different time points and pertussis responses at week 15. And in the plot here, you can see that the alpha diversity of pertussis responders was significantly higher at day 4 to 7 of life compared to non-responders who had a lower alpha diversity in their gut microbiota. Again, for the sake of time, I'm not showing any plots, but we also found that community composition of responders and non-responders differed at week 1 of life. And in a DSEQ2 analysis, the bacterial taxa that distinguished pertussis vaccine responders and non-responders best was Bifidobacterium cantinulatum, which was increased in pertussis responders. So to sum the findings of our study up, we did not see any stark differences in breast milk microbiota of women with and without HIV and their infants. We found little relatedness between maternal breast milk and infant gut microbiota, and mode of feeding had the strongest influence on the infant gut microbiota composition in our small cohort. We have some preliminary data suggesting that gut microbes in early life might influence vaccine responses in later life. And the question arises whether Bifidobacterium cantinulatum could have the potential to improve responses to parenteral vaccines, which is like an interesting way to follow, since this could be explored as a therapeutic avenue. So, one could hypothesize that administration of probiotics or Bifidobacterium probiotic during early life to HIV-exposed infants might improve their vaccine responses. So as a next step, we are measuring more pertussis vaccine responses in infants who have matched day 4 to 7 stool microbiome data to see whether this association holds up. And with that, I'm going to finish, and I would like to thank my collaborators. The study was funded via UO1, led by Catherine Blish, and Susan Holmes from Stanford University provided data analysis support. Sonwa Bielitz-Zanibe from the University of Cape Town measured the pertussis vaccine responses. And most, I would like to thank the clinic and the lab team and all of the participants. Thank you.
Thank you very much, Anna, for that wonderful presentation. I might also say that I know you're not feeling well, so we are really grateful for having taken the time to actually come and present this work to us. So thank you very much, Dr. Anna. Right. Before we go to the break, I think there are a few questions that I'd like to give to the presenters. Maybe we can answer them on the chat because of time. Prof. Pasmo, with the issue of funding being a challenge, how is the consortium able to bridge this gap when you're talking about Africa? Because that's what you try to put across, that we're trying to bridge this gap. So we have this issue of funding across the African countries or African research groups. So how is this consortium how will it be able to bridge this gap based on the fact that we also have issues to do with funding? So before you answer that, let me also give ask all the questions, and then let's see how we go about it. And then to Lauren Martin, do microbiomes from other body sites such as the rectum and the mouth influence the composition of vaginal microbiome and vice versa? And then to Anna Happel, do ARVs play a significant role in microbiome diversity? And somebody asked, I may have missed this, but does the mode of feeding take into consideration breast milk fed babies who received milk via bottle feeding instead of directly on the breast? Or do you mean those babies were formula fed? Thank you. So those are the three questions, one for each of you. Thank you. Let's start with you, Prof. Passmo.
Hi. So, I mean, that's the $1 million question, literally, or the R1 million question, being in South Africa, because obviously the research often follows the funding, and our initiative has been funded through a global health initiative from the Gates Foundation under Calesta Schumer African Leadership Fellowship. And the intention is that the funding is always going to be an issue on the continent because of funding issues. But having said that, several African countries have significant amounts of funding available for research. And I think coming together as a critical mass of experts with centers of excellence around this really creates like a sense of confidence in the research we're doing on the continent, that we keep each other in check, that we're using best practices, and it will make the research being done here internationally competitive. So although there is more funding coming from the North, countries on the continent are actually investing a significant amount of funding towards research that we really need to acknowledge. But also I feel that if we package ourselves strategically as a consortium working together, it creates the confidence, and the expertise is unified, that it will make fundraising more achievable. But having said that, I do feel that we'll continue to get good funding from the global North for this kind of research, because it's really, really important. And what my sense is, and why I think a leadership fellowship is really a perfect opportunity, it's negotiating better positions in publications, because my sense from working with many different collaborators from different countries is that the willingness is there, but sometimes I think as African researchers we don't not fight our corner, but we don't put our names forward for leadership positions, and when we do, it's reciprocated and accepted. So it's just about negotiating, more strategically. I'll stop there.
Oh, okay, thank you. Yeah, what you have said is actually quite right. I think we also need to actually put ourselves forward if we are to lead some of these projects. I think I'll go straight to Dr. Anna Happel, where we also had a third question. Now, I guess the other one you'll respond to it on the chat.
Sure. So there was one question whether we follow infants up for longer than 9 months. At the moment, we don't do that, unfortunately, because we only have funding to do it for 9 months. But I agree that it would be exciting to do a more long-term follow-up to look at infectious and non-infectious morbidity and growth outcomes. And then the other question was regarding, to clarify what we included in the mode of feeding. And so this included all infants who still received breast milk, no matter whether they received it directly from the breast or whether moms, expressed breast milk and fed it via a bottle. But we basically compared infants who still received breast milk for those who are exclusively formula fed.
Okay. So I'd like to thank all of you very much for having taken this time to give this presentation. Thank you very much. So before we go for the break, I'm sure we have eaten a lot of the break time. There are 3 posters which will be shown in the first 5 minutes of the break, and the 3 breakout rooms will be open thereafter for anyone wanting to chat in that time. And then also I'd like to mention that there'll be posters on display with QR codes for further reading for a while, and 3 breakout rooms for anyone who is interested. Thank you very much. Over to you, Boke.
So great. Thanks everyone for just bearing with us despite the interruptions. I think it's been a very informative and educative first 2 sessions. If you noticed, we had quite a bit of gut microbiome and oral microbiome presentations, perhaps indicative of the amount of research being done in those aspects across the continent. But I want to encourage us to take advantage of the breakout rooms right after the poster presentations or the poster slides going through. And also, there's this kind of aim to be back here at about 40 minutes on top of the hour. That is in about 13 minutes if possible. So that will be at 4:40 p.m. Central African time. So let's just target to be back at that time, and we will proceed with the next session, which promises to be exciting as well. So thanks again for joining the first half, and please stay on the call actually, because since we're trying to monitor the interference, we're being very restrictive in the waiting room session. So please just stay on the call if you're able to. If not, just make sure your name is on when you're joining in after the break. Thanks again, everyone, and see you in a bit. See you in about 12 minutes. All right, thanks.
So it's my great pleasure to welcome everyone back from the short break. And I thoroughly enjoyed reading the posters. And I just wanted to congratulate the team for the little QR codes as well that provided the nice link to the posters if you didn't have time to read through them. So congratulations to those who put posters forward. So I'm going to be chairing the next session, which is looking at respiratory and skin microbiomes. And I really wanted to extend my gratitude to Dr. Nadu, who has at very short notice agreed to give us a presentation today. On the program, we have our esteemed colleague from Stellenbosch University, Grant Turon, who sent us a message very late on Sunday evening, humbly apologizing that he's not well and unable to give the presentation today. So I am very pleased to introduce Dr. Nadu to talk to us about respiratory microbiomes. And I just wanted to check with her whether she is able to share her presentation.
Hi, Joanne. Just about to share now. Do you see it?
Yes, that's perfect. Thank you very much.
Awesome. Well, thanks very much, Joanne, for the introduction. And yes, Grant does send his sincere apologies. And yeah, so I'm going to be telling you all a bit about what we've been doing in the microbiome space regarding TB. So many of you may already know this, but TB has killed more people than any other infectious disease in history. And unfortunately, it continues to be a leading killer, especially in South Africa. More people die of TB than any other cause. And we also know that about a quarter of the global population are infected, although latently, with the bacterium, that's Mycobacterium tuberculosis. But interestingly enough, only about 5 to 10% will actually go on to develop active disease. And yeah, so we are still trying to understand the underlying mechanisms for why this happens in only a small subset of individuals. And TB is curable. It takes about 6 months for an infected patient to be cured. However, once a person has been treated for TB, they have a lifetime risk of getting it again. And so really within the CLIMB group that Grant Turon heads, one of the central themes of our research is on looking at the microbiome's potential role in these various stages of the TB spectrum. And yeah, so there's been a lot of emerging research in this sphere. And it's quite interesting that even though TB mainly affects the lungs, the gut microbiome is increasingly getting a lot of attention because there seems to be some sort of crosstalk that occurs between the gut and the lung, specifically concerning the microbes and the metabolites that they produce. So certainly from the animal models, we've seen that aerosol challenge with Mycobacterium tuberculosis results in these rapid changes that occur in the gut, even though MTB itself is undetectable in the gut. Also, if mice are infected or naturally colonized by a specific strain of Helicobacter or genus of Helicobacter, they show poor control of TB disease, as well as heightened pulmonary inflammation. And this was an interesting finding by researchers at UCT, where TB treatment itself depletes these non-tuberculous mycobacterial NTMs, which have been shown to have protective immune effects against MTB, and we wonder whether these NTMs play a role in why people get TB over and over. And then there's also been research showing that specific metabolites or compounds that are made by the commensal gut microbiome have or aid in MTB kill. So a lot of the research that I'm going to present here is embedded in many of the ongoing diagnostic investigations that are done within CLIMB. So the study that I'll be speaking a lot about is one of the more mature ones, where we've comprehensively described these patients as well as their close contacts. So in this study, it involved 4 groups of people. The first 2 groups of people were people that presented to the clinic with TB symptoms, and after us performing diagnostic evaluations, we were able to classify them as either a TB case or a symptomatic control. And then we also invited close contacts of each of these groups to participate in providing microbiome samples. And I'm going to focus on the stool results from these groups. So just to pay attention mainly to the red dots and the green dots, and what these show us, or what this plot is showing us, is what beta diversity looks like before these patients initiate TB treatment. So if we look at cases versus symptomatic controls, we see that they have very different gut microbial composition. And that was quite surprising to us because antibiotics have not even been introduced at this stage, but there are already gut microbiome differences between these symptomatic individuals. And then when we looked at this on a taxonomic level, we saw that within the cases, there were enrichment with these 3 gut anaerobes, and these anaerobes are significant because they are the main producers of short-chain fatty acids, which we've heard about earlier. So basically the way to understand this figure is bacteria that appear further to the left or to the right and further away from or further above the red dotted line are most discriminatory. And so we also collected blood from the cases and the symptomatic controls. And this network is just focusing on the cases. And we see that each of these 3 enriched anaerobes that I've mentioned correlated with distinct inflammatory immune pathways, which we know are associated with TB disease. So this study was really laying the foundation for how the microbiome compares, even pre-treatment, between cases and symptomatic controls. And we see that there is, in fact, a distinct gut microbiome that's specific to TB disease. So just to talk about short-chain fatty acids, so we know that these are made by specific gut microbiota as they ferment the dietary fibers. And in general, short-chain fatty acids, specifically the 3 main ones, butyrate, acetate, and propionate, are well, they have a beneficial role in respiratory disease in terms of dampening the inflammatory response, but we see a opposite picture in TB, and so I'm going to just speak a bit about that. So this is work that was done quite a while back now by one of our longstanding collaborators at New York University, and this work involved HIV-infected individuals who were followed up for the development of TB, and they were relatively healthy because they were stable on ART. And what they had done in this work is perform a bronchoscopy procedure, which is basically sampling the lower airways. And they found that these individuals were enriched in these lung anaerobes, as well as having high levels of short-chain fatty acids. And they found that when they added these short-chain fatty acids at ex vivo, in ex vivo assays to PBMCs, that they inhibited TB kill. And they also found, using this independent Cox multivariable model involving interferon gamma and IL-17, that if you had to double butyrate, that this was associated with a almost 6-fold increased risk for the later development of TB. So we can see that short-chain fatty acids don't have such a good role for TB, at least. And so going back to the patients that I discussed, this is work done by Dr. Samantha Modley, who's a postdoc in our group, and she measured the short-chain fatty acids in a subset of those patients. And we see already, based on this pilot data, that there tends to be an increase or higher level of propionate and butyrate in cases compared to symptomatic controls. And then I also wanted to bring in some of the interesting work that our PhD student, Georgina Anyawo, recently completed. So she submitted a thesis now. So this involves patients who had presumptive extrapulmonary TB, and in this case, it involves TB of the lymph nodes. And it was interesting to see that the microbiome in that space, in the lymph fluid, differs by TB status. And she was able to characterize the microbiome composition into these 3 microbial community states, which we termed lymphotypes. And of these 3 lymphotypes, the third lymphotype, DL3, which was dominated by Mycobacterium, was associated with increased predicted levels of short-chain fatty acids, as well as more severe disease based on the clinical characteristics of these patients. And then I also wanted to talk about treatment, because many of you will know that TB is one of the very few diseases that requires prolonged treatment with antibiotics. Some of these are broad spectrum. And if you're a patient who's diagnosed with drug susceptible TB, you're looking at consuming about 180 broad spectrum antibiotic doses over those 6 months. And then that, of course, increases in more drug resistant forms of TB. And so we decided to follow up the patients that I spoke about earlier, the TB cases, to see what the effects of treatment were. And so we collected sputum and stool from each of about 114 cases during treatment. So that would be month 2, the end of treatment, that's month 6, and at 2 time points post-treatment. And we also followed up their close contacts to have a sort of in-house control. And I'm not going to spend much time on this, but what I want to point your attention to here is that you can see in both sputum and stool, within the first 2 months, there is this rapid reduction in alpha diversity. And in stool, this persists until the end of treatment. But we do see that by at least 6 months post-treatment, that there seems to be a return to baseline levels in terms of alpha diversity. But in terms of beta diversity, here again, we see the clear shift from baseline to month 2. There doesn't seem to be a further shift between month 2 and month 6, but still different to baseline. And this perturbed microbial state continues all the way until 1 year post-treatment, at least in stool. And when we looked at this on a taxonomic level, again, we saw that if you compared patients at baseline to month 2, and this was done in a paired way, that treatment depleted 3 gut bacteria, specifically Akkermansia, which is part of the core gut commensals and is therefore associated with health. And it was also interesting to see at the end of treatment, we compared patients who had culture converted, so were considered, a successful outcome, and this would be the culture negatives, compared to patients who remained as culture positive for TB at the end of treatment, and saw that those who did culture convert had about a 30-fold enrichment of this bacteria called dialister, which we are now busy looking at on a deeper level. And then just
1 minute left.
Okay. So really what I just want to bring across here is we are close to finalizing a new method. This is in collaboration with Jose Clemente of ICANN School of Medicine at Mount Sinai. And what this method, or the idea behind this method, is to be able to eventually prognosticate outcome. And so this longitudinal data has been great for validating or at least getting that method at the point of publication. And so using this data, we saw that depending on how patients' alpha diversity fluctuated over time, they could be clustered into different groups. And the 2 groups of interest here are cluster 1 and cluster 4. Cluster 1 comprises patients who've never had TB before, whereas cluster 4 solely comprises patients who had at least 1 episode of TB before, or were treated for TB before. And so, I just wanted to touch on that. And I'm nearly done. So this slide is just to show some of the other longitudinal studies that we have ongoing in our group, and we are completely open to collaboration. There's plenty of room for that. And so I encourage you to chat to me afterwards. And yes, I would like to acknowledge our amazing, passionate team, excellent team, almost like family, as well as all of the funders. Thank you.
Thank you. Sorry, I cut you off a little bit abruptly, but what I really wanted to have is just a moment for any direct questions. Selfishly, I'd be really interested in reaching out to you. I'm really intrigued by the impact of drugs on the microbiome, but also at different sites to the lung and GI tract. So I really found those findings really important, and I will send an email.
Awesome.
Is there one short question? If not, I am going to go on to our next 2 presenters. And if there are any questions about this exciting study, please put them in the chat. And I just urge you to possibly answer them in the chat as well, in case we don't have additional time for more questions. But thank you very much for that really interesting, thought-provoking presentation. So our next presenter is Jessica McClintock, who will be talking to us about nasopharyngeal microbiome samples from infants in Zambia with RSV. And over to you, Jessica.
Good afternoon. My name is Jessica McClintock, and today I'm excited to share with you the results of our nasopharyngeal microbiome RSV study. As many of you probably know, RSV is the most common cause of bronchiolitis and lower respiratory tract infections in children in the first year of life, disproportionately affecting infants in low and middle income countries, as shown here on the map. Previous studies have found that the nasopharyngeal microbiome of infants with RSV infections has specific characteristics that correlate with severity of disease, including lower biodiversity, perturbations in the microbiota, and differences in relative abundance. These studies have focused on infants seen in a clinical or hospital setting, predominantly in higher income countries. We therefore wondered if similar correlations would be found at an extreme end of severity of disease, namely death, in a community setting in a lower income country. Our team previously collected nasopharyngeal samples as part of the Z-Prime study in Lusaka, Zambia. In this study, each decedent underwent one-time post-mortem nasopharyngeal sampling. These samples were each tested for RSV, and then we randomly selected 50 RSV-positive deceased infants and matched them by season of death and age of death to 50 RSV-negative decedents. This provided a group of 100 infants between the ages of 4 days and 6 months old. We first observed the overall abundance of the microbes in RSV-negative and RSV-positive samples, which can be seen in the stacked bar plots here. I want to draw your attention to the trend that we see represented by these lines, where the microbes below the line represent the 4 most abundant microbes in RSV-positive samples as listed on the slide. The different trend that we observe between the two groups is an encouraging sign that there are in fact compositional differences between the nasopharyngeal microbiomes of the RSV-positive and RSV-negative decedents. To better elucidate what these differences are, we next looked at differences in diversity. We used 3 different metrics to analyze alpha diversity and found no significant difference in diversity across these metrics. Next, we turned to beta diversity analyses. We computed a Bray-Curtis dissimilarity index and visualized these results using non-metric multidimensional scaling, or NMDS, and principal coordinate analysis, or PCOA. In the NMDS plot A, we see a cluster of RSV-positive samples to the bottom left. We also see some distinct clustering of RSV-positive samples in the PCOA plot in plot B at the bottom right-hand corner. We completed Permanova and found that there was a significant difference in the centroid and dispersion of the Bray-Curtis dissimilarity of p of less than 0.001. This indicates that the diversity of each RSV-positive sample is more similar to the diversity in the other RSV-positive samples rather than to the RSV-negative samples. We already established that the difference in diversity is not due to the type or abundance of microbes present. Therefore, we compared the differences in relative composition of each microbe between RSV-positive and RSV-negative samples in a differential abundance analysis to determine if relative abundance accounted for this finding. This resulted in 3 genera being statistically different, namely Gemella, Moraxella, and Staphylococcus. These genera are shown here as box plots to indicate the different abundances between groups. We see that Gemella and Staphylococcus have lower abundances in RSV-positive samples, and Moraxella has higher abundances in RSV-positive samples when compared to RSV-negative decedents. Previous studies have shown that infants with RSV have decreased abundances in Gemella and Staphylococcus and an increase in Moraxella abundance. Our study supports these findings. Our study is limited in the fact that this is a cross-sectional study. Our results may be confounded by antibiotic exposure since we have no records of potential antibiotic use in these infants. Our study adds to the body of knowledge on RSV since very few studies look at RSV in infants at this extreme end of the spectrum, namely death in a community setting. In conclusion, this all points to the fact that we do indeed see differences in the microbiome at this extreme end of RSV severity in a community rather than a hospital or clinical setting. Gemella, Moraxella, and Staphylococcus are all differentially abundant between our groups. This naturally leads us to wonder if the change in microbial presence precedes RSV infection or if it is an effect of the infection. We believe that the microbiome is different prior to infection, causing some infants to be more susceptible to RSV. This could be studied further in a longitudinal study that follows infants from birth through contraction and potential recovery of RSV while taking samples at multiple time points to compare the development and change of the nasopharyngeal space leading up to and after the infection. Being able to identify infants with a greater susceptibility to more extreme forms of RSV early in life would allow medical teams to provide better and earlier care to reduce the severity of the disease. I'd like to thank and acknowledge the Bill and Melinda Gates Foundation as well as NIH for funding this project. I would also like to thank our team and all the work that they put into this project, especially Dr. Johnson, Dr. Gill, and Audrey Odom. Thank you.
Thank you for that excellent presentation, Jessica. And I think in the interest of time, if there are questions for Jessica, please pop them into the chat. She is online and will be able to answer. And our last presentation from the session is changes in nasopharyngeal microbiomes and Streptococcus genus during influenza, based on a cohort recruited in South Africa. And the presenter is Maimuna Karim. So over to you.
Hi everyone, I am Maimuna Karim, a scientist working at the NICD in South Africa. My talk today will be giving some of our preliminary results of the changes in the nasopharyngeal microbiota and Streptococcus genus during influenza infection in a community cohort study in South Africa. My presentation will have the following outline. Disequilibrium in the nasopharyngeal microbiota can occur due to acquisition of new pathogens, resulting in a proliferation or overgrowth of a pathobiont and dissemination leading to disease. One such example is the interaction of influenza with the pneumococcus. The synergistic relationship between influenza and the pneumococcus has been documented and dates as far back to the 1918 influenza H1N1 pandemic. Investigations into the 1918 pandemic indicate that many patients were infected with a secondary bacterial infection, which led to an increase in morbidity and mortality during this pandemic. Further evidence suggests that viral infections are associated with elevated pneumococcal density and invasive pneumococcal pneumonia. And animal models have also shown that a prior influenza infection could enhance the transmission of the pneumococcus. This suggests that the introduction of influenza into the nasopharynx disrupts the microbiome equilibrium, leading to an overgrowth of the pneumococcus, thus increasing the transmission and dissemination of the bacteria to the lungs, causing pneumonia. We therefore aim to understand the changes that occur in the nasopharyngeal microbiome as a result of influenza infection, looking particularly at the Streptococcus genus in a community cohort study in South Africa. This study was nested within a prospective household cohort study of influenza, RSV, and other respiratory pathogens, community burden, and transmission dynamics in South Africa, known as the FIRST study. Individuals were enrolled each year for 2016 through 2018, with a new cohort enrolled each year. Briefly, 327 households were enrolled at 2 community sites. One was an urban site and the other a rural site. We enrolled approximately 100 households for each year, and swabs from individuals were collected twice weekly for 6 to 10 months. For the microbiome analysis, a subset of samples were randomly selected from the 2017 cohort. 16 srRNA sequencing was performed on 534 samples. These samples comprise 69 controls and 452 samples from the first study. After sequencing, we performed alpha and beta diversity analyses. Alpha diversity was assessed using Shannon's diversity index, and beta diversity was done using Permanova. Statistical differences by influenza infection time period was calculated using a linear mixed effects model controlling for age and repeated measures. So a median of 43,532 reads were generated per sample, which were binned into a total of 1,084 ASVs after filtering, representing 16 phylum, and these are represented in figure 1. Proteobacteria was the most abundant phylum at all 3 time points. Figure 2 shows the relative abundance of the top 10 ASVs by study period observed in the nasopharynx. The overall spectrum of these genera appeared to be slightly different between the 3 groups. This figure shows the alpha diversity using Shannon's diversity index by influenza infection time period. We observed that the alpha diversity differed by age. However, after adjusting for age and accounting for repeated measures, we observed no statistical differences in the alpha diversity over the influenza infection time periods. This figure shows the baseline nasal microbiota composition in association with influenza period by using a non-metric multidimensional scaling plot, where each plot represents the microbial community composition of one sample. The samples were colored by sampling point in relation to an influenza period. We detected a significant difference in the beta diversity across influenza periods, which remained significant after adjusting for age. The last slide of my results shows the patterns in relative abundance of top 6 genera in participants before, during, and after an influenza episode. After adjusting for age and accounting for repeated measures, we observed that the diversity in the Streptococcus genus did not differ by influenza infection time period. In conclusion, we observed differences in the overall microbiota composition, but these were not translated in changes in the 6 major genera, including Streptococcus. Further ASV level analyses are required to further pinpoint the origin of community level differences. That brings me to the end of my presentation. I would like to thank the following people and entities for all their assistance and financial support, and a special thank you to the participants for agreeing to be a part of the study. Thank you.
Thank you for that. And we're over time, and we don't want to be rude to Professor Gordon. So I'm going to hand straight over to Volker to do the introduction. And any questions for Dr. Karim, please include them in the chat. Thank you to our panelists in this session for excellent presentations, and looking forward to your presentation, Dr. Gordon.
Fantastic. Thank you so much, everyone. Please stay till the end of the presentation because I am so excited, very humbled, very privileged to be introducing somebody I met very recently in person, and I was just blown away. And a person that actually needs no introduction, Professor Jeffrey Gordon. He's the Dr. Robert J. Glazer Distinguished University Professor, Director of the Edison Family Center for Genome Sciences and Systems Biology at Washington University School of Medicine, St. Louis, Missouri. I'm not going to spend much time going on because I think his name is pretty much synonymous with microbiome research. And I think they want to hear less of me and more of you right now, but I'll go ahead and just say that today he'll be speaking to us about developing microbiome-directed therapeutic foods for childhood undernutrition. And at the end of his talk as well, he will just share some of his experiences on microbiome programs and just developing this kind of capacity in different parts of the world. Please, over to you, Dr. Gordon. Thank you.
Well, thank you so much for this opportunity. It's a pleasure to be here today, albeit remotely. I want to talk about a shared journey that has involved a group of people who've been longstanding, inspiring collaborators at the International Center for Diarrheal Disease Research in Bangladesh, led by Tamid Ahmed, and our group here at Washington University School of Medicine. The focus has been on childhood undernutrition, the leading cause of death of children under 5 worldwide. Lots of epidemiological studies, as you all know, have shown that it's not due to food insecurity alone, that many factors operate within and across generations. Current therapies have reduced mortality, but alas and alack, they've had very limited success in ameliorating the long-term sequelae of undernutrition, including failed linear growth, persistent stunting, immune dysfunctions, neurodevelopmental impairments. So we're missing something in our understanding of pathogenesis, and we certainly don't have a comprehensive definition of the biological state or states of undernourished children. These children are identified based on anthropometry, with the definitions well known to all of you of what moderate and severe stunting or moderate and severe wasting are. So we're pursuing a hypothesis that there is a definable normal program of gut microbial community assembly that begins at birth, and that this process is disrupted in one or more ways. And such a disruption in community assembly is not simply an effect of undernutrition, but a contributing cause. The corollary to the hypothesis is that there is a well-choreographed program of co-development of the gut microbiota and host systems and subsystems that is an important underpinning of healthy growth of infants and children. So to test the hypothesis, as you know, requires a multi-step journey. First, to define in the population where the burden of disease is great and where you ultimately want to do an intervention, what normal is. And then to develop metrics to quantify deviations from normal to see whether those deviations from normal are correlated with disease prevalence and severity. If such correlations are found to be significant, then a test of causality has to be performed, and hopefully emerging from the preclinical models that might be initially used for such tests of causality are thoughts about therapeutic targets. And following such an identification process of these targets, a deep amount of thinking about the type of therapeutic strategy that would be employed. It would have to be culturally acceptable, affordable, and scalable in this set of conditions. So to define normal, when this program began, Tamid, together with his colleague Saeed Hook, and also a very talented MD PhD student in the lab, Satish Subramanian, took a look at the developing fecal microbiota of children living in this urban slum in one of the districts of Dhaka. The slum is known as Mirpur, and the trust, the devotion of the healthcare workers to the community is enormous, well established, and the commitment of the mothers to the health of their children profound. So this allows long-term sampling to be performed of given individuals, and it's an ideal setting for birth cohort studies. So such a birth cohort study was undertaken in Mirpur with monthly fecal sampling beginning at birth, initially through the first 24 months, but extending to 60 months. And at the time, our approach was to look at domain bacteria and to define which taxa were present at what proportional representation using 16S rRNA gene amplicon sequencing. So what Satish did was to apply machine learning algorithms to that large data set, but limit the analysis to those children who had consistently normal anthropometry, i.e. children who had manifested positive deviance. They had escaped undernutrition, which is so prevalent in this community. So 50 members of this birth cohort that had consistently normal anthropometry over the 5-year period of time were subjected to this machine learning driven analysis to identify age discriminatory bacterial taxa. So here in this heat map, each row represents one of these taxa. Each column represents the monthly age bin. We're going from 0 to 60 months. And this provided an opportunity, at least to initially describe what might be normal. So if you took a vertical slice through this heat map and looked at a given age bin at the proportional representation of these age discriminatory strains, you could operationally define a state of maturation or development for the microbiota among these biologically unrelated children living in a common region. And that was done. But we thought that it was relatively insufficient because, as you all know, microbes don't exist in isolation in the communities that inhabit our various body habitats. There are complex interactions between these microbes as well as between the microbial community and the host. So Arjun Raman was a very talented postdoc in the lab at the time, and he took the statistical tools which were developed in the field called econophysics. Econophysics have been applied to a variety of different systems, but principally look at covariation of features within systems, including systems like the stock market in the United States. So are there covariations in stock prices, for instance, that could be correlated with the activity of different economic sectors? But that principle was applied to these children's gut communities. And Arjun asked whether there was a small group of covariant bacterial strains, she termed an eco-group, that could describe the normal development of the microbial community in healthy members of this birth cohort. The metaphor is basically, if you were to try to understand the emergent and overt behavior of a flock of birds, looking at individual birds in that flock in isolation wouldn't tell you what the pattern, the dynamics, the organizational features of that flock would be. And the same thing for ants foraging, etc. There are lots of examples of this type. So Arjun used this group of 15 taxa to describe further the developmental biology of the gut microbiota in this cohort. And he found that the community assembly process defined in this way was largely completed by the end of the second postnatal year. But that children who had undernutrition had microbial communities that looked younger than you would expect based on their chronological age. And a number of studies were done together with Tamid, looking at children with SAM and with MAM who had been treated with standard therapy for their undernutrition, therapy that was not developed based on a consideration of or knowledge of the developmental biology of the microbiota, and found that with these therapies, these interventions, this immaturity was not substantially repaired. So these children were left with persistent microbiota immaturity. Question is that an effect of the undernutrition, or is it a contributing cause? So having defined normal in this fashion and developed a metric for quantifying deviations from normal and seeing that this younger appearing immature microbiota was more characteristic of children with undernutrition, particularly more severe undernutrition than in milder forms of undernutrition. A test of causality consisted of introducing microbial communities from the target population of interest into young germ-free animals, fed the diets of that population. And the donors are either healthy with normally maturing microbiota or undernourished with immature microbiota. Again, the donors were chronologically age matched, and for each fecal sample, well preserved. In fact, the protocol in Bangladesh is to, within 15 to 20 minutes after production of a fecal sample, to put it in these dry shippers that are cryogenically charged. So it's liquid nitrogen temperature, and to keep them in those shippers until they can be put at -80 degrees, and that -80 degree cold chain is preserved until samples are analyzed in various ways. So these microbial communities are then introduced, each donor's community, into a group of young germ-free animals. And the test is to what extent are phenotypes of the donor transmittable to the recipient gnotobiotic animals. And this test of causality revealed discordant phenotypes between mice that receive undernourished donor microbiota as opposed to mice that received healthy donor microbiota. And the discordant phenotypes involve lean body mass gain, which could be quantified using quantitative magnetic resonance, changes in bone growth, which could be quantified in part by microcomputed tomography, as well as looking at serum biomarkers of osteoblastic and osteoclastic activity. Metabolic function in different tissues is defined by targeted mass spectrometry, and various aspects of immune function, including barrier function. So the tests of causality indicated that perhaps the microbiota in these children had effects that operate within and beyond the walls of the gut. And the task became to use this preclinical model where animals were colonized with the communities of the various children who we'd ultimately want to treat, to identify targets. These targets were identified using machine learning algorithms, and specifically looking at all the microbial communities that have been transplanted to all the groups of animals. A series of organisms were designated with machine learning algorithms to be growth discriminatory. That is to say, their representation was correlated with certain aspects of growth, including weight gain, lean body mass gain, etc. These organisms identified in this fashion, these bacterial taxa, it turns out were not appropriately represented in the immature microbiota of undernourished children. In a number of cases, they were underrepresented and presumed to be underperforming. and they became our therapeutic targets. We cultured these organisms in a number of cases, sequenced their genomes, added these organisms to immature microbiota, and saw that if that addition was done prior to transplantation into germ-free animals, you could ameliorate some of the transmissible phenotypes that I alluded to before. So what kind of therapeutic strategy or concept would we use based on these preclinical studies? A consortium of cultured genome sequence growth discriminatory strains as a next-gen probiotic was interesting to consider, but impractical, both from the standpoint of manufacture, but also cost. So we decided to turn to food, and particularly to test the hypothesis that certain complementary foods, the foods to given between cessation of exclusive milk feeding and a fully weaned state, might contain nutrients that would be coveted by these growth discriminatory organisms, and that we should screen, as Jeanette Gehring, a PhD student in the lab, together with a postdoc, Sid Venkatesh, and another PhD student, Hao-Wei Chang, did. We should screen these complementary food ingredients in notobiotic animal models, harboring the very microbes from the population that we wanted to treat, in order to create either single ingredient or multi-ingredient formulations that we would call microbiota-directed complementary foods that could be used as agents of repair of the children's gut communities. So that was our strategy. This is how the strategy was played out. This is a complicated slide, but we basically start out with food ingredients, complementary foods, culturally acceptable, available, and generally affordable, introduced into the diets of germ-free animals colonized with a consortium of age and growth discriminatory strains from this population of children living in Bangladesh. And that individual food ingredients would be screened, with the initial readout being changes in the fitness or abundances of the targeted organisms that we wanted to manipulate. And this was done with single ingredients initially, and also then with combinations of ingredients. Along the way, whenever we would get a hit, we would ask ourselves, are these leads from sustainable sources based on current or envisioned agricultural practices? Are they affordable? And how about food processing considerations? Again, this is early on in this discovery process. We wanted to make sure that the foods would have satisfactory organoleptic properties, that is to say, taste, texture, and smell. So the first generation of microbiota-directed complementary food prototypes emerged from these studies of mice colonized with defined consortia of age and growth discriminatory strains. And the effects on the consortia, not only in terms of fitness, but also gene expression among the community members, and also certain aspects of host biology were assayed. The leads from this screen were then forwarded to another type of screen in which we would take the intact, uncultured microbial communities of children with undernutrition who had been given standard therapy. These children's microbiota would have this persistent microbiota perturbation, this immaturity, as it were. And we would ask whether the lead MDCFs coming out of the first phase screen would be able to repair the microbial communities towards a healthy state in this preclinical model. And the answer was we did have leads emerging from this second screen. We looked at the effects not only on the microbial community, but on different aspects of growth in the mice. And from that set of experiments, round two screen, as it were, we had a series of leads that we could either directly advance to a pre-proof of concept study in humans, representing the population whose microbes were put into our preclinical model, or we could pause and take a sobriety test and ask ourselves, we're trying to change the developmental biology of children. That could be very productive, promote their growth, but there also could be important safety considerations. So rather than going directly from mice to humans, perhaps we should select a second host species whose physiology and metabolism are similar to that of humans, more similar to that of humans than mice. And that was a decision that was made. It was inspired by these concerns, but also produced a lot of perspiration because a lot of work was done, particularly by Hao-Wei Chang, to develop a protocol that was very robust for rederiving piglets at birth as germ-free animals and maintaining them in notobotic isolators so that in the first month of their postnatal life, we could introduce the microbial community members of these children into piglets and feed them diets that contain our lead MDCFs. And from that second type of host species experiment, the third round of our screen, emerged several different MDCF candidates. So a nutritional rehabilitation center was established by our colleagues at ICTRB in Mirpur in the slum, and a pre-POC study conducted of these MDCF prototypes. The children were 12 to 18 months old. They had MAM, moderate acute malnutrition, with weight for length z-scores of -2 to -3. There are 4 arms in the study, 15 children per arm. 3 different MDCFs were tested together with a ready-to-use supplementary food, which was a common or standard food supplement used to treat children with MAM in Bangladesh. There was an enrollment period. There was a run-in phase where there was fecal sample collection, anthropometry, and at the end of the 2-week run-in phase, a blood draw was obtained so that we could look at the levels of 1,305 different plasma proteins representing many different biomarkers and mediators of different aspects of host physiology. We wanted a comprehensive definition of the biological state of these children prior to the treatment phase, because ultimately we saw that there's an opportunity to connect dots, that as we reconfigure the microbiota, it would be potentially possible to link changes in the representation or express functions of different components of the community with different aspects of host systems physiology. So the intervention twice daily feeding under supervision of each of these MDCFs, 1 MDCF to a particular group, weekly fecal collections, and at the end of the 4-week period of time, anthropometry, a blood draw for this aptamer-based proteomic analysis using SomaLogic's technology, and then a 2-week follow-on period. And from this analysis emerged the following results. There was one clear winner. One of the MDCFs was a clear winner based on its ability to repair the microbiota, definable by the change in representation of those eco-group taxa. It also is the clear winner in terms of changing the levels of plasma proteins that were associated with healthy growth. So we had a reference data set of the plasma proteomes of healthy children living in this area, as well as those children chronologically age-matched who had SAM, severe acute malnutrition. So we selected a MDCF prototype that did the best job among the 3 in repairing the microbiota and the best job in evolving the plasma proteome during this 4-week period of time to a more healthy state. We did not anticipate, and nor did we find, that based on the powering of the study, 15 per arm, and the length of treatment, that we would see a statistically significant change in anthropometric measures of ponderal growth, WLZ, and certainly not on LAZ, the marker of linear growth. So this is a nutritionist view of the MDCF that we selected. And it also is a nutritionist view of RUSF. It's not necessarily a microbial view. So the principal complementary foods in MDCF2 were chickpea flour, peanut flour, soybean flour, and green banana. There is a major source of lipid, and that is soybean oil. And there are minerals and vitamins added as a micronutrient premix. RUSF, in contrast, has rice and lentil. It does have powdered skim milk, unlike MDCF2, and its principal lipid was soybean oil. I'd like you to look at the bottom on the left column and see the caloric densities of MDCF2 compared to RUSF. MDCF2 had 15% lower caloric density than did RUSF. So the stage was set for a randomized controlled clinical trial of MDCF2 versus RUSF in 12 to 18-month-old children living in Mirpur with MAM. And the stage had been set, again, by developing this MDCF using the microbial communities of these children in this set of preclinical models I described. So the design of the trial is a 3-month intervention. At time 0, we have a blood draw, a fecal sample, and anthropometry. A week before, we have anthropometry and a fecal sample collection. Either RUSF twice a day or MDCF2 twice a day under supervision, initially in the clinic. The second month, 1 day under supervision in the clinic, and 1 day, 1 time a day, once a day at home. And the last month, twice daily supervision at home. But throughout the period of treatment, the 3-month period of treatment, fecal samples were collected on a regular basis. The blood draw was at 1 month and at 3 months. Then we would pause and do something that we thought was very important. We would do initially, and we being Tamid Ahmed and Ishita Mustafa, who were largely responsible for the execution of this study, there was going to be a 1-month follow-up. But we made a secret pledge to ourselves that we would commit ourselves and apply to the IRB for a 2-year follow-up. And the importance of that I'll describe at the very end. So the primary outcome measure of this trial was a change in WLZ, and the secondary outcome measures was microbiota repair and plasma proteomic biomarkers and mediators of growth. So this is the results that we reported in 2021 in the New England Journal of Medicine, that the rate of change in WLZ was significantly greater. Increase was significantly greater in children receiving MDCF2 compared to RUSF. And I just want to go back for a second and just emphasize the powering of this study. There were 62 children enrolled per arm. 59 made it in each of the arms. So this was not a huge study. The children who received MDCF2 showed significantly faster weight gain than those who received RUSF, even though MDCF2 is 15% less calorically dense than RUSF. So you might ask the question, what is the underlying mechanism for these effects? So Robert Chen is a very talented MD PhD student in the lab, and SomaLogic had advanced its technology. The platform now could measure 5,000 protein simultaneously in using less than 100 microliters of plasma. And for each child in both arms of the study, all 118 children, the rate of change in their WLZ scores over time were correlated with the change in levels of each of these 5,000 different proteins. And 70 proteins were identified that had statistically significant positive correlations with WLZ. And they included a number of known mediators of musculoskeletal development, CNS development. There were also proteins that were significantly negatively correlated with WLZ, and they included primarily biomarkers and mediators of inflammation. Very intriguingly, MDCF2 produced a significantly greater increase in these 70 proteins members of the set of 70 proteins that were positively correlated with WLZ, and a significantly greater decrease in the levels of proteins that were negatively correlated with WLZ. So I'm not going to go into these proteins in detail because I don't have an enormous amount of time for this talk, but I wanted to turn to the microbiome and talk about work that Matt Hibbard, Daniel Weber, and Hannah Lin did. And that was to search for WLZ-associated bacterial strains. So in this particular case, what was done is to look for MAGs, high-quality MAGs, in this population of children who had either received MDCF2 or RUSF. So deep shotgun sequencing of the fecal microbiomes, serially sampled for each participant prior to, during, and after treatment. And we had on average 9 samples per child. So each child's fecal DNA samples underwent shotgun sequencing, and each child's reads were assembled into MAGs. At the end of that process, we dereplicated all MAGs from all individuals and got 1,000 high-quality MAGs and used linear mixed effects models to identify those MAGs that were correlated with WLZ. So the results of this analysis are shown here. The results revealed 75 different MAGs that were significantly positively correlated with WLZ. And the reason we wanted to go through this process of MAG assembly is a lust to be able to look at not only the abundance of MAGs as a function of treatment, but also what genes were they expressing, what metabolic pathways were they expressing as a function of treatment with MDCF2 or RUSF. Could we get an idea of what the targets were of MDCF2? And could we get an idea of what the bioactive components of MDCF2 were as a result of this type of analysis? So for all the MAGs that were WLZ correlated, in silico metabolic reconstruction was done using the microbial community seed paradigm. There are 106 different pathways involved in carbohydrate utilization, amino acid metabolism, biosynthesis, vitamin biosynthesis, intermediary metabolism. This was a scheme that was developed and reported by Andrei Osterman and Dmitry Rodionov. So what was noted early on by Matt Hibbard when he did this type of reconstruction with Andrei and Dmitry's help was prominent enrichment of pathways involved in utilization of various complex polysaccharides by these WLZ-associated MAGs. At this stage, set the stage for an analysis using microbial RNA-seq of differential gene expression, and also an analysis that would correlate differential gene expression, particularly of these pathways involved in carbohydrate utilization, with knowledge of the glycans present in MDCF2 versus RUSF. So we've had a long-standing relationship with Carlita Labrilla, and for all of the food staples that were introduced into MDCF2 and to RUSF, the glycan landscape was defined by ultra-high performance liquid chromatography, triple quadrupole mass spectrometry. The comparisons of those analyses of the food staples, as well as the formulations themselves, MDCF2 versus RUSF, revealed distinctive representation of different polysaccharides in one versus the other formulation, including a enrichment in galactans and mannans in MDCF2 versus RUSF, and some of the structures are shown here. These are the glycans that are represented. So this is a microbes view of a therapeutic food formulation. And this also represents the intersection between microbiome science, food science, and human nutrition. The microbes sense the chemical repertoire that's represented or embodied in a particular food formulation and respond to that by trying to access the nutrients contained in that formulation. They don't see the labels. They don't see chickpea, soybean, peanut, or banana. So this helps enormously if you have these structures in hand, interpreting the microbial RNA-seq analysis. So the same total nucleic acid preparations that were generated from each fecal sample from each individual in both arms of the study were used, on the one hand, to purify DNA for the shotgun sequencing and MAG assembly, but also to isolate RNA for microbial RNA-seq. And Daniel Weber, using the models described on the left of this, looked at carbohydrate utilization pathways that were differentially expressed in the microbiomes of children receiving MDCF2 versus RUSF. And you can see that the types of pathways that came up from this analysis are targeted towards those glycans that are enriched in MDCF2 compared to RUSF. And if you take a look, as Dan did, between those children who received MDCF2 and were in the upper quartile of WLZ responses and those that were in the lower quartile, and compared and contrasted the patterns of gene expression in those two groups or subgroups, he was able to get even higher resolution views of what the bioactive components of MDCF2 were. An equally remarkable result was that the differential expression of these carbohydrate utilization pathways in MDCF2 treated children were linked, mapped onto, to a substantial degree, to just two MAGs, both assigned to P. copri. They weren't the only MAGs that these reads mapped to, but they were the principal MAGs that these reads mapped to. So two MAGs among the 11 MAGs that we had assigned to P. copri appeared to be the originators of this transcriptional response to MDCF2. So that allows us to look at WLZ response, expression of genes in these pathways, two levels of microbial products of MDCF2 glycan metabolism as judged by using the same fecal samples that were used to generate fecal DNA for shotgun sequencing, RNA for microbial RNA-seq, triple quadrupole mass spectrometry of the glycans that are there as a function of treatment. And that gives us an idea of what the bioactive components are. And that's going to be very important, not only for ultimate decisions about dosing, but if we were to create biosimilar MDCFs. So the analysis of what the bioactive components of MDCF2 were based on not only the MCC pathways that I alluded to, but also the polysaccharide utilization loci that are represented in P. copri. So I said that there were 11 P. copri MAGs that were identified in this population. The two that were the originators of these transcripts were the only two that were significantly positively associated with WLZ. The rest were not significantly associated with WLZ. And these two MAGs contain pools that were very similar to one another. This group of pools shown here, colored in green, consists of pools that were functionally conserved and structurally conserved between the two MAGs. And the yellow is functionally conserved, but somewhat distinct. The degree of amino acid sequence identity to the encoded carbohydrate active enzymes in these pools, and the pools I'm alluding to are MAGs 18, pools belonging to MAGs 18 and MAG 19. The yellow ones aren't as conserved, but you can see the higher degree of conservation of pools in these two MAGs compared to all the other MAGs, none of which the other P. copri MAGs were WLZ correlated with. And we can predict the glycan targets of these pools based on the known, experimentally determined substrate specificities of the carbohydrate active enzymes encoded by these pools, or bioinformatic analysis of what the specificity should be. And this is work that has been done together with Bernie Henrichon, a long-standing collaborator who is a world expert in carbohydrate active enzymes and who maintains the KZ database. So that helped guide our analysis of the glycans in fecal samples, but also the MAG genome features help guide our culturing efforts. So because the fecal samples have been so well preserved, we could go back to them and start culturing out age and WLZ associated organisms that correspond to the age and WLZ mags that we had identified. And that effort is summarized here in this overly complicated slide, but I will draw your attention to strains of P. copri that we cultured that are highlighted in red. The mags are in blue, and we get cultured isolates of P. copri like BGF5_2, very difficult to pronounce nomenclature. And you can see the degree of conservation of pool in this isolate compared to the pools that were conserved in mags 18 and 19. It turns out that over 100 of the 106 metabolic pathways that had been annotated by MCC were also conserved in this particular P. copri isolate. So we could test using this isolate in vitro what its carbohydrate utilization phenotypes are, and how these phenotypes could be correlated with the glycans in MDCF2, because we could obtain purified glycans that are represented in MDCF2 from commercial sources and test their ability to support growth of P. copri in a defined media, in this particular strain of P. copri or other strains of P. copri. That sets the stage for the final experimental slide that I'm going to show you, which is a reverse translation experiment to validate the importance of these P. copri mags. So the experimental design involves a female germ-free animal, a dam, who gives birth to a litter of pups. And the female mouse is first colonized with a consortium of cultured, genome sequenced, age and WLZ associated bacteria. Initially, groups of organisms that are early colonizers of the intestines of children living in this area. That consortium, once received by the dam, is transmitted to her pups. The pups are consuming at this stage breast milk exclusively. Consortium 2 is not really a consortium in this particular instance. It consists of the P. copri strain that we had cultured that most resembled MAG 18 and 19. And consortium 3, which was introduced as these animals were weaning, and the weaning diet that they received was similar to the weaning diet of the study participants. And that consortium 3 consists of later stage colonizers. So you have a circumstance where you are colonizing in a sequence that resembles community assembly in the population. The children are receiving a sequence of diets, breast milk exclusively first, then a weaning diet similar to the study participants, and ultimately are weaned onto MDCF2. And there's only one tweak that you should know. Half the animals received all 3 consortia, and half the animals we omitted consortium 2. That is to say, they did not receive P. copri. So we can compare and contrast the biological properties of the animals that receive an identical sequence of diet and all but P. copri in their defined community versus those that did experience P. copri in their community. And the results of the comparison of these 2 groups of animals is that mice that lack P. copri had a different weight gain. In fact, the ones that had P. copri had significantly greater weight gain. The ones that had P. copri had markedly greater processing of MDCF2 glycans as defined by mass spectrometry. And using single nucleus RNA-seq of the intestine, we see marked effects on energy metabolism in different gut epithelial cell lineages involving lipid metabolism, amino acid metabolism, carbohydrate metabolism, plus the expression of different nutrient transporters. Changes we saw with single nucleus RNA-seq, which were validated by targeted RNA-seq. And the spatial patterns of changes in expression that are brought about by the presence versus absence of P. copri are really exquisite and involve principally the enterocytic lineage, and particularly enterocytes positioned at the base and middle regions of villi. So this is work done by Hao-Wei Chang, Yi Wang, Evan Lee, and Kaylee Pruce. So I'm going to conclude and then take a look ahead for a second. The conclusions are that this is, we think, some evidence from clinical studies, reverse translation studies, that healthy growth is linked to healthy co-development of the gut microbiota. We have in this particular population of 12 to 18-month-old Bangladeshi children identified microbial therapeutic targets, shown that it's not just calories, and emphasized the importance of glycans as critical bioactive ingredients in MDCF2. And knowledge of these glycans is an important factor ultimately when considering dosing strategies in the future, and also ingredient sources, which cultivars you might choose that could have equivalent glycan landscapes. It's critical that we advance this to other populations, which we are doing, tests of generalizability, durability, and long-term safety. It's important to consider age as a variable. Remember the central hypothesis about co-development of the microbiome and host and healthy growth. It's important to determine how malleable the system is, how plastic the system is. So for instance, does the duration and severity of disrupted community development prior to the intervention affect responses to the intervention? How about the chronological age when the developmental disruption of the community first occurs? These are really important things to test in a formal way. And of course, the World Health Organization will want to have a therapeutic intervention that won't be limited just to 12 to 18 months, but also could be expanded to 6 to 24 months. And of course, it's also true that there may be such devastation of the gut community that food-based interventions will be insufficient to repair that community. Long-term follow-up, exclamation point, bold font, maybe even italics, very important. And although we haven't reported this, one interesting result is that I had mentioned to you about the significant association between levels of certain regulators of musculoskeletal development and WLZ when we first did the proteomic analysis. And although we didn't see any statistically significant difference in LAZ at the end of the 3-month intervention, we did see that emerge in the 2-year follow-up. So we're currently, together with the support of the Gates Foundation and WHO, engaged in the Nutrimam study, which will test the generalizability of MDCF2's effect on the microbiome and growth responses, again, in the context of MAM. This is a study design, 6 to 24-month-old children with MAM, plus or minus acute illnesses, a total of 1,200 participants per site. Those sites are in Bangladesh, India, Pakistan, and 2 sites in Africa, Tanzania, in Dar es Salaam, in the Temeke district, which has a population of 1.1 million inhabitants, as well as in Mali, which will focus on a community health center-based intervention. It'll be a peri-urban area in Tanzania. And the first phase of the test, 3 months in duration, is focused on anthropometric recovery. There'll be 3 arms: locally produced MDF, RUSF, and locally available foods. And then the follow-up phase would be either, and it's 3 months in duration, an intervention package, which consists of counseling and a food voucher, or standard care, which is routine care and counseling. There's going to be a mechanistic sub-study in both Mali, in Tanzania, and Bangladesh, where in the first phase, we'll use fecal samples that are collected as well as plasma to do the types of analyses that I just described for the randomized controlled study of MAM in Bangladesh. And the phase 2 study would be to look at the durability of microbiome repair, which would involve fecal samples as well as the blood samples. So aptamer-based proteomic analysis, shotgun sequencing for looking at MAGs, and microbial RNA-seq if possible. The importance of these mechanistic sub-studies is to identify, as many of you talked about, important biomarkers and mediators of host responses, in part for stratification of the populations prior to intervention, as well as for evaluation of efficacy, including the durability of the effects of microbiota repair, and ultimately later adaptive clinical studies. We want to have a better definition of wellness, and also to use this information to have more informed policies about complementary feeding practices, to connect the ingredient profiles of complementary foods to healthy development of the microbiota. Voki wanted me to spend the remaining 4 hours that was allocated to me, just kidding, about some thoughts of establishing microbiome programs in Africa. And I just want to spend 5 minutes max to talk about this concept of human microbial observatories at sites in selected low and middle income countries where long-term sampling of microbial ecology in a population could be conducted. And I think it's important to think about all of the changes that are occurring in the world, both environmentally and also epidemiologically in terms of changing patterns of disease, whether we could identify a series of sites where long-term sampling could be initiated and perpetuated, and where the information gleaned could help direct efforts at development and testing of new therapies for treating those diseases. So these are the thoughts that I'd like to throw out for consideration. First of all, how do you design such a program? And I think it's important that the sites selected within a given country or sites selected have a population that exemplifies a global health challenge or challenges of interest, that the anthropologic features of the population are exemplary for that particular global health challenge or challenges of interest, and that there might be a prediction that these features of the population might change over time. It's also important to consider how the environments where the population lives are predicted to change over time. And given the enormous impact of climate change, one can contextualize these environmental changes at these sites, and also contextualize how considerations of environmental changes and the anthropologic features of the population would direct the habitats, both human and environmental, that could or should be sampled. Community engagement and trust, obviously critical for longitudinal studies. I gave you examples of what was required in our work in Bangladesh. So the level of trust that would exist at a site that would be considered for these microbial observatory centers, what kind of trust exists between the healthcare workers and community leaders at the site? What's the history of community engagement? What is the likelihood that participants can be recruited successfully and retained in birth cohort studies and other types of longitudinal studies that require follow-up? Early considerations of how governmental regulatory policies would impact current and future microbiome research. And there are many dimensions to this. What about policies related to sharing of biospecimens and metadata? What about the regulatory policies about prebiotics, probiotics, and symbiotics? What about the sourcing, approval, and distribution of microbiome-directed therapeutic foods that represent foods that were created within the country using cultivars from the country or cultivars that are imported? What is the general belief about how these foods would be available, could be available, should be available? Another consideration for the design of such a program: develop and implement best practices and policies for ethical informed consent that deal with issues of ownership of microbes and future use provisions for commercial and non-commercial uses. So how can these consents be obtained from the population in an ethical way, in a way that everybody is informed? Are there early thoughts, because I think they're critical, about equitable and realistic policies for economic benefit to local and national industry? Because this has to do with the consequences of success in a microbiome analysis that might lead to new therapeutic targets and therapeutic concepts. So how would IP stewardship occur in an equitable way? What's the government's attitude towards this? Can you work closely with relevant government agencies in developing such policies? How about decision-making about which body habitats to sample at what ages? If you're doing these microbial observatory programs, even if they're well aligned with disease, how would you go about sampling? You have an infrastructure and strategies to preserve biospecimens to ensure the long-term viability of component organisms, including also the metabolic products of these organisms, and the capacity to retrieve these stored samples in a cost-effective manner. A decision-making tree about which samples, if you do capture them and preserve them adequately, would you characterize, both by culture independent and dependent methods? And what's the time frame for doing so? These collection activities scale very rapidly and become very costly. It's important, given that you align these decision-making trees, I believe, with diseases of interest, cost considerations, the partnerships you might have both with commercial and non-commercial firms, and also what is envisioned or what might exist currently for therapeutic lead development programs. And then finally, branding. If there is a microbial observatory program established in a given country at a site, or maybe in some cases more than one site, the principal observatory site should be branded a regional center of excellence. Why? That could help drive capacity building for microbiome research, not only at that site, but also at other sites within the country, as also potentially if there's cooperation between countries. And then leverage these centers to promote data exchange between centers and sites where microbial observatories are established, using harmonized policies for exchanging that data. And also leverage the centers to promote clinical studies with lead therapeutics to determine their efficacy, generalizability, durability, safety. And finally, the last slide, brand the microbial observatories as decadal programs, not just 1 or 2 years, decade-long programs to obtain sources of sustained funding, including from programs that are very much centered on climate change and environmental sustainability. So those are my thoughts about considerations that might underpin the development of what I'm naming microbial observatory programs within countries. So I'll end with 2 words, thank you, and I'll repeat them again. Thank you for this opportunity. I really appreciate it.
Wow. Thank you so much, Dr. Gordon. And this has been absolutely fantastic. I mean, not just the talk about the translational aspect of microbiome research, which we all look and hope our research, heads towards at some point, but also just the suggestions for building these microbial repositories, which really looks like a fantastic idea. I just feel so bad that this was at the end of the day when people actually have dinner, supper in many parts of Africa, and they've missed out on this amazing presentation. But thankfully, they will have a chance to access them. But I am not going to spend the whole time talking because there are a few questions for you in the chat, and I will just go ahead and read through. I mean, raving comments in the chat section. But the first question was from Professor Kwedi from Tunisia. She asked about, I have to scroll all the way up. She says, thank you, dear Professor Gordon, for this excellent presentation and for the new insights. I wanted to know, if it hasn't been done already, if we could consider implementing this personalized protocol for treatment response in pediatric cancers.
I think that all of this deals with a central issue in the field. What is your therapeutic target? What do you know about mechanism of action? And how can you build an estate of knowledge about the efficacy of a particular intervention? So the temptation in a field such as ours is to get a lead and to start testing it very widely and against many different pathologic states. But we really have to determine first and foremost, let's say if there's a pressing global health challenge that's prioritized, whether a particular therapeutic concept works. How well does it work in terms of the generalizability? How durable does microbiota repair occur? How durable is that repair? And what are the effects of microbiota repair on host biology? It's not that we shouldn't have broad ambition, but I think that we need to have the gift of attention. The gift of attention is something that only we can give ourselves, and to establish the concept that microbiota-directed therapeutics work and work in the ways I allude to. Undernutrition is many different biological states, and I think that there'll be many different microbiotic configurations that will influence pathogenesis in variable ways. So it itself is a huge area to delve into. And children are dying every day. So you want to try to move the testing forward as quickly as possible. So implementation and scaling is also a huge part of the consideration. And how do we get these therapeutic food formulas distributed to areas that exemplify the burden of disease and test efficacy? That's one of the purposes of NutriMap. And will organizations like the World Health Organization create a set of data requests that we can satisfy in order for them to consider a change in policy? Most of the formulations currently designed to treat severe acute malnutrition, SAM, these RUTFs, are not designed explicitly to repair the microbiome. So how do the policies of UNICEF, Codex compliance, all sorts of things, relate to the development of a microbiota-directed therapeutics? And can these formulations comply to existing requirements, or are you going to have to change the formulation in ways to satisfy existing requirements in ways that reduce efficacy? So there are all sorts of things to consider. That's a long-winded answer to your question, which is we need to know targets, mechanism, and to move forward thoughtfully in an experimental medicine pipeline, but with rapid translation to the population.
Excellent. Thank you so much. I know our time is far spent and you've stayed on. Would you have time for one more question, perhaps, to Gordon?
Absolutely.
Great. Just to say, everybody, you can feel free to put on your switch on your cameras so we can see you. You know, there are real people behind the names. So this is from Professor Pasmo. She did present a bit earlier. She says, this is inspiring, exciting to see translation of microbiome research into health-changing interventions. Wondering what infant uptake, that is user experience, was for the MDCF2 compared to RUSF. Also, whether host changes to intervention were noted. Particularly interested in gut barrier function, proteins or similar. She's quite inspired by this research. She did say, it's actually in the chats as well. So she did say that Also, sorry if she missed this, but what was the background food consumption like in infants? Did additional food intake influence the effect of the MDCF2?
Okay, so we did not note a. It's a great question, by the way, and all of us bang our heads against the wall when we try to figure out what children are consuming based on food frequency questionnaires. There was not a significant difference in food consumptions as defined by FFQ between the RUSF treated group and the MDCF treated group. In Bangladesh, partial breastfeeding continues through 18 to 24 months of age, I should just say, in case I'm aware of that. We did see a hint that the children in the upper quartile of WLZ response who had received MDCF2 compared to those that had lower quartile had a slight increase in the consumption of legumes in their background diet, legumes that could contain glycans, similar to those that are represented in MDCF. I'm a huge believer in trying to find other ways of quantifying food consumption, and they may be available with DNA-linked methods to look at the representation of plant DNA in the fecal samples that are being sequenced, and a variety of other approaches. But I think it's critical. There's a food encyclopedia that Carlito Labrella has generated, which correlates the major world's food staples with the glycan content of those food staples. So if we think about food from a microbes perspective, is what are the chemical ingredients, not just the labels, what are the biochemical components of these foods, and how does that relate to responses and differences in dietary patterns? Sorry, that wasn't a clean answer, but maybe that's sufficient to generally address what was raised.
I think I'm muted. So I just want to say that there are so many questions popping up now, and I think our time is far spent. We're like about 20 minutes over the hour. And just as maybe I'm just going to read out the questions. Feel free to answer or not to answer, depending on your time. I just want to read them. I don't know if you can see the chat, but there are 2 questions actually. Actually, no, tell you what, I can send this across to you and you can send email responses if that's okay, because I can see that.
Okay, I think that might be the best thing to do. I agree. I unfortunately going to have to go in a few minutes. So
that is perfectly fine. Just to say a big thank you again to you and all the presenters and all the attendees. This has been fantastic. And I do apologize again for the interruptions we had earlier, and that made us kind of restrict access to the meeting a bit. So that might have affected the numbers as well. Just to say that this has been beyond my expectation. I'm very grateful for everybody that responded to our invitation, both last minute and ahead of time as well. And just to say that normally the idea of this is generally to foster collaborations, networking across the continent and across everybody working on African microbiome samples. And that this platform is to just showcase the research regardless of the stage of the research. As you could see that the different research projects are at different stages, and just to encourage everyone, come show us what you're doing. I mean, that's the whole point of this. Let's see what you're doing. Let's see what others are doing, so we can see areas of synergy and how we can work better together, because I'm pretty sure that collaboratively we can achieve a lot more and more impactful research. Just to say a few thank yous to, again, everybody that has attended, the speakers, both invited and abstract submitters. Without you, this would not have been possible, and we are so grateful. My profound thanks to the organizing team, you know yourselves, and to the H3Africa Coordinating Center for just sticking with us. Just to say again that this definitely is not all-encompassing of African microbiome research, but thanks to those that submitted, but we'd like for a wider diversity, so we can actually see the spectrum of the different research across the continent, across the different sites being worked on the continent as well. And we hope that this creates a platform for networking and potential collaboration. Reach out to me if there's somebody you noted or you don't have access to. Reach out to the coordinating center. We'll find a way to connect you. To this end, we also have the H3Africa Microbiome Task Force. We meet every month, every third Monday at 4 p.m. Central African time. The idea is to form collaborations, networking, and just to see how we can better build capacity. We've also extended this to, it's evolving now to the African Microbiome Special Interest Group. This group meets every second month together with the the H3Africa Task Force. Also, every second month, also on the third Mondays at 4 p.m. You're encouraged to join. We have a Slack group where we try to keep everyone updated on what is going on. And just say a big thank you again to everyone. Thank you so much. See you next year, hopefully with more submissions and more diversity in this research. And again, thank you so much, Dr. Gordon. I'm not going to call you Jeff. I'm sorry. I have to call you Dr. Gordon or Prof. Gordon.
Please, I wish you would.
Thank you so much to all the speakers again. We're very grateful, and have a lovely evening, day wherever you are at this time, whatever time it is for you, wherever you're in the world at this time. So thank you so much, everyone, and have a good evening. Bye.
Bye-bye.
Bye-bye.